Well, hello everybody. How's everybody doing out there? Happy Cinco de Mayo. I'm so happy that you're on board. And my name is Sam Turco. A few introductory comments about myself. I've been a professor of biochemistry and medicine at the University of Kentucky Medical School for many, many years. As far as Kaplan Medical, this is my 17th year of doing these reviews for Kaplan Medical. And in terms of these live online, I've been doing these live online since the beginning that Kaplan has been doing the live online. So I feel that I'm a veteran. I've seen a lot of glitches and things and problems, and but we'll overcome them. We'll overcome them. And if we have any kind of glitches, so any case, I'm so happy to be on board for the next 11 days. Now that what I specialize in, you're looking at it, and that's biochemistry. Now I know, because I'm a veteran, I know a lot of medical students fear biochemistry. They despise biochemistry. They see it on the schedule and they say, oh man, 11 days of biochemistry, this is going to be awful. I have no idea why biochemistry is not one of your favorite disciplines, if not your favorite disciplines. It should be your favorite discipline, because if you understand biochemistry, biochemistry explains pathology, it explains pharmacology, it explains micro and these other things that you have or will have after me with Dr. Harris in pharm and Dr. Barone in pathology. If you understand biochemistry, a little bit of biochemistry goes a long way. Now, the reason why I think a lot of medical students hate biochemistry is it was never taught to you correctly. It was a lot of students, a lot of faculty show you structures and pathways and all this memorizing this and memorize. They don't understand it. That's my goal in the next 11 days. That if you're here, let me stress that if you're here right now and you expect me to go through a lot of memorization issues, a lot of mnemonics. Hold on. Sorry about that. Forgot to. I'm a veteran. I know what a phone call sounds like. Disturbing. But the idea is that if you're into mnemonics, if you're into memorization, that kind of stuff, I'm telling you right now, you're in the wrong place. You're in the wrong place. If you expect me to give you the mnemonics for the urea cycle, the Krebs cycle, that kind of stuff, forget about it. You're not going to get the mnemonics from me. You can get that in first aid. It's all in first aid. You can get mnemonics for, and so what do you need me for? What you need me for, and what I specialize in, is that if you're into big pictures, understanding, if you're into integration of biochemistry with all the other disciplines, if you're into explanations of why you give a certain drug to treat a certain disease caused by a certain microbe, you're in the right place. Because that's what I love to do, is talk about big pictures, understanding what goes on. Because if you understand, if we make sense of what's going on in the human body, things will come alive. Oh yeah, now I know what that pathology is about. Now I know Cushing's syndrome. Now I know diabetes so much better. Now I know why you give metformin. Because if you understand a little bit about chemistry, it goes a long way. And that's what we're going to do in the next 11 sessions. Just give me a chance. That's all I ask. Just give me a chance. If you never understood biochemistry, don't fear it. Give me a chance to explain it. And that's my goal, is to tell you what you need to know and what you don't need to know. And there's a lot of stuff you don't need to know. Matter of fact, let me stress here as well. A lot of people tell you that you should read ahead, later chapters, so when the instructor is talking, you know what they're talking about. In my opinion, I go against the grain. I would not read ahead. Because if you read ahead, you might be reading about something, you might spend a whole hour reading about the lac operon and how the microbes turn on gene expression, and I'm going to come up here and say, lac operon, low yield. Don't worry about it. And you wasted all that time. So in my opinion, don't read ahead, because you might be wasting valuable time. My opinion is, after the lecture, after the 4 hours of lecture, then go back when you have time, as soon as you can, go back over the high yield items. I will stress to you what's high yield, what you need to know, what's high yield, what's medium yield, and what's low yield. Stuff you don't have to know. I'll point it out to you, what you need to know and what you don't need to know. And your job, as long as I do my job properly, explain why it's high yield, as long as you do your job and go back and master the high yield material. Every session, there's going to be a lot of high yield material. I don't want to waste your time. We're going to go through a lot of high yield material. That's what you need to master. That evening or the next day, go back over it, over the high yield material. And if you have questions, ask me. I'll give you my email a little bit later. But the idea is, understand. That's what I'm trying to get you to do. I'm going to do everything I can in these 11 sessions to try to get you to think and not memorize, to get you to understand big pictures instead of doing mnemonics. I've never encountered a medical student who became a better medical student, became a better doctor because they know the mnemonics for the Krebs cycle. You're wasting time. But understand what these cycles do and so on. That's what's critical to know. So that's what we're going to spend these 11 sessions on. And right at the very end, when I'm done a week from Friday, my 11th day here, right at the very end, it's my goal that after we go through all this stuff, a lot of stuff coming your way, you say to yourself, I'm hoping you say to yourself, I'm glad I came. It was worth it. That's my goal. Not that I'm not asking you to love biochemistry, but I'm hoping that you benefit. You benefit from understanding. You master a lot of stuff, and you're going to do extremely well on board exams. If I do my job properly, explain. You do your job properly and pay attention. Don't go texting people while we're talking. Pay attention. As long as you pay attention to those type of things, you're going to do well on board exams. Because I will tell you what's high yield and what's low yield, what you need to know and what you don't need to know. That's my job, and I take it very serious, what you need to know. All right, now another item here is, the biochemistry lecture notes. What it is the first 7 chapters deals with molecular biology, and that takes about 2 to 3 sessions to go through molecular biology. And then after that, we're going to skip to do medical genetics, which is at the end of the book, because it makes sense. After we go through all that molecular biology and DNA and all this kind of stuff, it makes sense to talk about chromosome issues. And that's medical genetics. So we'll spend about 2 sessions on medical genetics. And then the rest of the time, we'll go back to biochemistry, chapters 8 through 18, which is the rest of the time, and we're going to go through all the metabolic stuff and get into a lot of big pictures. And you will see, I'm hoping, if you're paying attention, you give me a chance, you will see, oh yeah, now I understand. Oh yeah, that makes sense. Makes sense of what's going on. And it's so much to your advantage. Be positive. Don't be negative. Don't be negative about biochemistry. Be positive. What's in this discipline that's going to help you out? That's what you need to draw. And I'll tell you what you need to know and what you don't need to know. Now, the way I do my reviews is I scan the figures from the book. Everything I do is on PowerPoint. I write nothing, none of that chicken scratch stuff. I don't do any writing, all this kind of stuff. It's all, I don't use my hand and then underline things. Everything I do is on PowerPoint, animation. It takes a long time to do the animation. But that's the way I do it, is animation. And so what I do is I scan the figure from the book, and at the lower right-hand part of the figure, the lower right-hand part, if you'll see a number, that is the page number where the figure is located. So if you come in late, or if you're sleeping and then you wake up and say, oh, where is this guy? All you got to do is look at the lower right-hand part. That's the page number where the figure is located. So that's the way I do it, and then I add things to the figure. And now another thing I do in my reviews is it's a two-way street. So I can't see you, but you can see me. But I need to interact with you. I need to know whether you're following me and things like that. So what I do is the public chat pod is always up right below the camera, right below the video there. It's always up. And please, I'm going to ask you a lot of questions coming your way. A lot of questions, oral questions, and please type in, use what you know. Type in responses. Now, the way I want you to do this is type in your response and then look at the public chat pod and see how your response compares with your other classmates. Don't look at the public chat pod first. Type in your response. If you give the wrong answer, it's not a problem. Don't worry. If you give the wrong answer, no problem. We learn by our mistakes. But you have to participate. You're asking me, what's the best way to study? And the best way is to participate. You can't sit back and think you're going to take in all this information and spit it back out on the USMLEs. No, you have to participate. You have to be active in responding. And if only a few people respond, that does you no good. You have to respond. And if you give the wrong answer, we learn by our mistakes. We learn by our mistakes. My wife tells me that all the time. We learn by our mistakes. She's always correcting me. Just make sure she's not here. We learn by our mistakes. So please type in responses. Now, if a lot of you may have looked at videos and things like that. And if you have this stuff in front of you, if I ask you what's in, look at the videos and you have the answer in front of you, don't respond, because I'm looking for people who can recall things from other disciplines, things along that line. So again, participate. Please participate, and you will see, trust me, that you will start to understand. Things will start to make sense if you participate. Here. Okay, so that with all that stuff out of the way now, okay, all that preliminary stuff out of the way, what we're going to do is, Eamon, please go slow. I can't go. I got only 11 days. Remember, this is a review. It's not the first time you've had this stuff. I'm trying to go through at a certain pace so I don't bore students. At the same time, I can't go too fast or too slow. I just have to go at a steady pace because this is a review, not the first time you've had this stuff. So I'll tell you, I'll do the best I can, but you have to keep it, you have to pay attention. I'm telling you right now, if you go away because you got a text message or you're going to answer something and you come back, you're going to be lost. You got to be paying attention. Don't worry about other distractions. Pay attention, okay? And you're going to do well. All right. Now, with that in mind, we're ready to roll. So what we're going to do is we're going to get into molecular biology for about 2 to 3 days. And in chapter 1 is a somewhat boring chapter. And the reason is, in chapter 1 is nucleic acid structure and organization. In other words, in this chapter, we're going to talk about and review what is DNA, what is RNA, what is a nucleotide, that type of stuff. And so it starts off as a somewhat boring chapter, but please bear with me for this first hour. Things start to get a lot more interesting, but we must cover the basics. You'll never understand chromosome stuff unless you know what DNA is, and you'll never understand DNA unless you know what a nucleotide is, what a nucleoside is, what a base is. That's what this chapter is, giving the fundamentals of stuff coming down the road. Okay, so now with that in mind, the first figure I want to talk about is nomenclature. There's a lot of nomenclature in this figure, a lot of terms that we need to make sure you understand the term. And here are 3 terms you must understand. And that is the term of replication, transcription, and translation. Now, for some of you, this is boring stuff because you've had it many times before. You're probably yawning at me. But for some of you, this may be you haven't had it for a while. It's okay. As long as you understand what I'm saying here is you must understand these words. The replication means taking DNA and making more DNA. The term of transcription means taking DNA, making RNA. And the term of translation means taking RNA and making protein from that RNA. These 3 terms, you must master them, On board exams, if they say to you, in transcription, a certain drug was used to treat some microbe, you got to know what transcription means. You got to know what replication means. There are drugs coming down the road that affect all three of these terms, and you must know what the terms mean. Replication, transcription, and translation. Matter of fact, we have a whole chapter. Chapter 2 deals with replication. Chapter 3 deals with transcription. Chapter 4 deals with translation. Each chapter, a lot of pharmacology, a lot of pathology associated with each of the three terms. You must know these terms. The sooner, the better. Master the terms. Now, you just had microbiology with Dr. Muscatello, and she talked about a term there called reverse transcription by the enzyme reverse transcriptase, which takes RNA and making DNA. Now, that does happen in certain microbes, retroviruses, but reverse transcription also takes place in the human body normally. And I'll stress, because the National Board asks this from time to time, I'll stress where in the body you have reverse transcriptase. But the idea is most of the time it is transcription. It takes DNA and makes RNA from that. Now, so chapter 2, 3, and 4 deal with each of these terms. Chapter 5 is another term that the National Board has been hammering medical students on. And that's the term called gene expression. How do you find a gene in your 46 chromosomes and turn it on? This term, gene expression, integrates. Why is it so high yield for National Board exams? It integrates with every discipline. Once we get to chapter 5, you're going to see the integration of biochemistry with pharm, path, micro, physiology, endocrinology, embryology, all coming up with gene expression. Molecular stuff coming up with all the other disciplines. High yield chapter. Chapter 5, modern molecular biology. Okay, now that doesn't come until tomorrow sometime. But before we get to that, though, we're going to start right at the beginning and talk about what is DNA and how does it compare with RNA. In other words, compare these two terms of replication versus transcription. Now, in comparing these two terms, replication and transcription, one thing is again, replication means DNA synthesis. Transcription means RNA synthesis. Now, before we talk about the differences between DNA and RNA in terms of structures and things, one thing I want to mention is that the sizes of the nucleic acids differ. The lengths of DNA can be different from the length of RNA. Now, what I mean by this is the first of many, many questions I'm going to ask you over the next 11 sessions. The first of many. Get ready with a public chat pod. Get ready with the responses. If I were to say to you that the DNA was taken from one of your chromosomes, the protein was removed, the DNA was then laid out in a linear arrangement. Suppose that DNA was stretched out and was this length. So if I take one of your DNAs, one of your chromosomes, get rid of the proteins, take the DNA in that chromosome and stretch it out. Remember, you have linear DNA. Your DNA is linear in contrast to bacteria, which have circular DNA. Your chromosomal DNA are linear. So if I took the linear DNA and stretched it out, suppose it would look like this. All right, here comes the question. What would RNA look like compared to that DNA? Again, don't go looking in the public chat pod first. You're not fooling anybody. You're not fooling anybody. Type in your response and then look at the public chat pod. What would RNA look like? Shorter, but how much shorter? How much shorter? That's right, well done. It would look like a little speck compared to that DNA. So then you ask, well, so what? The so what is, this has to do with chapters 6 and 7 in molecular biology. The idea is that when you want to analyze DNA or manipulate DNA, for example, when you want to do genetic testing of a disease, genetic testing for person identification, Genetic testing in forensics. How often, like if you watch NCIS on TV, you know, all those crime scenes, and they're talking about somebody's DNA is being analyzed. That my wife watches all the time, all these programs like NCIS, Miami, Chicago, whatever. But the idea is when you want to analyze DNA, because DNA is too large. How large is DNA, the chromosomal DNA? It's staggeringly large. If I took the 23 chromosomes that you got from your mother, 23 chromosomes that you got from your mother, and took the DNA from all 23 chromosomes and lined them up lengthwise. So I take 23 chromosomes that you got from your mother, took the DNA and lined them up lengthwise. You know how long that DNA would be? It would be a staggering 1 meter in length. Think about that. 1 meter in length is the chromosome, is the DNA that you got from your mother. 1 meter. So each of the chromosomes has the length of the DNA is very large. And so the idea is that when you want to analyze the DNA, you can't analyze it directly because it's too large. So what happens is the DNA must first be fragmented. You cut up the DNA first, and then the DNA fragments are analyzed. And in chapter 7, we're going to be talking about a very important, powerful technique called RFLP, restriction fragment length polymorphisms. So you can analyze DNA after the DNA is fragmented. The DNA fragments are then analyzed on some sort of electrophoresis, southern blots, all coming up a little bit later in chapter 7. Don't worry about it now, but right now just focus thinking to yourself, man, DNA is amazingly large. Whereas RNA and protein made from the RNA, they are sufficiently small and they are never cut up. Never ever cut up. You never cut up RNA or protein. You just had immunology with Dr. Muscatello in her great lectures. And in immunology, you never hear about protein being cut up in Western blots. In Western blots, you analyze the protein directly because protein is sufficiently small, it's never cut up. And RNA and protein, they're small, they are never cut up, never fragmented. Whereas DNA is always fragmented first. When you want to analyze the DNA, to analyze some crime scene or to analyze some disease like sickle cell anemia, we're going to be doing that in chapter 7. It'll be a family with a sickle cell trait. How can you analyze the DNA to determine the offspring, whether they are homozygous for sickle cell, heterozygous, or normal for the beta globin? All coming up later on in chapter 7 as a preview of things coming down the road. Hopefully you understand that that's all high yield stuff. Now another item here in comparing replication and transcription, and that is the phases of the cell cycle differ. The phases of the cell cycle in which replication takes place can be different than when transcription takes place. So that's what's coming up next, is the review of the cell cycle, the all-important cell cycle. You must understand the cell cycle. High yield cycle is the cell cycle. Now you may have talked about it in other disciplines, but the idea is here is G0. Now in G0, this is G1, S phase, G2, and M phase. Now cells in the body which are in G0 phase, these are cells that are resting, non-dividing cells. Let me see your answers here. Let me see your answer. What's an example, in case the National Board asks you, what's an example of a cell type in your body right now that's a cell that's in G0 phase? What's an example? That's right, you all should be getting this. Neurons are in G0 phase. Skeletal muscle cells, cardiac myocytes are in G0 phase. Liver cells are in G0 phase. In your body right now, you have approximately 10 trillion cells. Think about that. 10 trillion cells, give or take a couple cells here or there. 10 trillion cells. The vast majority of them as an adult are in G0 phase. So they're resting, non-dividing cells. But there are some cells that you must know that are in G1 phase. These are cells that will grow. So if the National Board asks you, what's an example of in histology or cell biology, what's an example of a cell type in your body right now that's in G1 phase that will grow? What's your answer? Let me see your answers here. What's an example? Not red blood cells. Red blood cells don't grow. If you said red blood cells, you're thinking nucleated red cells. Erythrocytes don't grow. Please be precise. So you'll never forget it. Those of you who said red blood cells, you'll never forget it's nucleated red cells. Because once you lose the nucleus, they don't grow. Erythrocytes don't grow. But nucleated red cells are in G1 phase. Other examples, epithelial cells that are in the skin, epithelial cells that line the GI tract, follicle cells, great examples of cells in G1 phase. Now those cells in G1 phase are sitting there waiting, and then eventually they'll go into S phase and go into G2 and M phase. Now in S phase, if the National Board asks you what occurs in S phase, you say, this is where the process of replication takes place, only in S phase. You got to know what that term means. Replication means DNA synthesis, right? So where do you do DNA synthesis of the chromosomal DNA? In S phase. So at the beginning of S phase, there are 46 chromosomes in a cell. At the end of S phase, the chromosomes have multiplied by 2. There are 92 chromosomes at the end of S phase. We'll talk a lot more about that later on in genetics, in karyotyping, and so on. But at the beginning of S phase, there is 46 chromosomal DNAs. At the end of S phase, there's 92 chromosomal DNAs. And then those cells will go into G2, and then the cells will divide in M phase. All right, now here comes a question for you. So if replication takes place in S phase, let me see your answers. Where does the process of transcription take place? Now, without looking back in the previous page, you got to know what transcription means, right? High yield. Where do you do transcription? And transcription means RNA synthesis. And the answer is, where do you do transcription? And it's throughout interphase. G1, S phase, G2 phase is where you make RNA. But there is no RNA synthesis, no gene expression in M phase. So if the boards ask you, where do you make RNA in the cell cycle? You say G1, S, and G2 phase, but not M phase. Now here comes another question for you. Now this is where you have to think. Again, I'm going to do everything I can to get you to think instead of regurgitation. Think. What about cells in G0 phase? Cells in G0 phase do not undergo replication. They don't multiply the chromosomal DNA. But do cells in G0 phase, do they undergo transcription? You got to know what that word means. Do cells in G0 phase, do they undergo transcription? That's right. Now this is a case where you can memorize. You can say, oh yeah, yes they do. But here, this is the way you think about this. This is the way you get to the answer without memorizing. Because what's an example of a cell in G0 phase? The liver cell, right? The hepatocyte. And you know the hepatocyte, they don't grow, but normally. But the idea is that me and you, our hepatocytes are making many proteins. They're making albumin, they're making transferrin, they're making all the blood clotting factors, and all those are proteins that are being secreted in the bloodstream. And so those are proteins. Proteins come from RNA. RNA comes from DNA. So the answer has to be a resounding yes. Cells in G0 phase undergo transcription and translation, but not replication. There it is. There it is. I mean, no reason to memorize. Don't put away the first aid book. You don't need it because you're a thinker. You're blessed with a beautiful mind. I'm going to get you to use that beautiful mind of yours. Use it. Use it. No reason to memorize. It just makes sense that cells in G0 phase undergo transcription, translation, but not replication. All right, now another item here. Major concept. I told you I love big pictures. Here's a big picture item. Cells in G1 phase to go into S phase. They do not automatically go into S phase. For cells in G1 phase, epithelial cells, nucleated red cells, and so on, to go into S phase, there has to be some signal, right? There has to be a signal that tells a cell in G1 phase, hey, take the 46 chromosomes and begin multiplying by 2. So what is it? What is the signal in the body? Do you think there's a fairy in the magic in your body right now? Do you think you're in Disneyland or Disney World? And what's that fairy's name? Tinkerbell. You think Tinkerbell then waves the magic wand and all the cells in G1 phase begin to multiply? Of course not. What is it in your body that tells cells in G1 phase? And the answer is it's a discipline called endocrinology. Because when Dr. Wilson was here at the beginning of the course, and he talked about all those growth factors like epidermal growth factor, PDGF, and so on. What do those growth factors do? What they do is they bind to a class of receptor called the tyrosine kinase class of receptor. I got a lot to say about that later on in biochemistry. The class of receptor are the tyrosine kinase class of receptors. And then when the hormone binds to the tyrosine kinase class of receptor, then that signals the cell, take the 46 chromosomes and begin multiplying by 2. The integration of biochemistry with molecular endocrinology. I got a lot to say about that later on. Tyrosine kinases, we talk about that later on once we get to insulin. How does insulin work? Works by tyrosine kinases as well as growth factors. A lot to say about that next week here. But when something goes wrong with the biochemistry, when something's wrong with the signaling, either with the receptor or the signaling cascade, then that's the course called oncology. Oncology that Dr. Barone elegantly, you're going to love Dr. Barone in a couple weeks from now in pathology. Dr. Barone's fantastic. But when he's here and he's talking about all these cancer cells and all these cancers and so on, it's just something going wrong with the signaling. There's cells going into S phase when they should not go into S phase in the absence of the hormone. That's what oncology is all about. When something's wrong with the signaling, cells in G1 phase now go into S phase when they should not in the absence of the hormone. And you'll never understand the oncology unless you understand the biochemistry first. That's all coming up later on in chapter 9, once we get to insulin and growth factors, how they signal a cell to start multiplying. All coming up a little bit later. All right. Now what's coming up next is the rest of the chapter is we're going to build up to what a DNA, what is a chromosome. That's what the rest of the chapter is about. What is a chromosome? And before we talk about what a chromosome is at the end of the chapter, we got to talk about what is the DNA in the chromosome. And to talk about what DNA is, we got to talk about the precursors that are used to make the DNA that's found in a chromosome. All right, so now that's what's going to come up. We're going to start at the beginning, at the basics. What do you need to know about the precursors that are used to make DNA and RNA that ultimately end up being the chromosome? So what's coming up next is what biochemistry is notorious for, and that is structure. Now, structure. And let me say something about structure. You're going to ask me, and rightfully so, do you need to know structures for the national board exams? In my opinion, that's low yield. I doubt. Nobody can guarantee anything. I cannot guarantee something will be on the board exams. I cannot guarantee that something will not be on a national board exam. All I can say is this is high yield, this is high probability, this is a low probability. And structure for boards is low probability, low yield. So don't go memorizing structures. But in my opinion, it's too late to memorize structures. So even if you wanted to memorize structures, it's too late for that. But in my opinion, much higher yield is you should know roughly what they look like. In your mind right now, you should know what glucose roughly looks like. You should know what cholesterol looks like. You should know what a fatty acid looks like without the gory details. That's what I mean. You should know roughly what these molecules look like, because if you know roughly, approximately what they look like, it goes a long way, as you will see coming up here with bases found in nucleotides used to make DNA and RNA. Now, bases, these are the bases you find in DNA and RNA. And these bases are classified into two subclasses. One class are the purines, and the other are the pyrimidines. And the purines have two rings. Again, don't go memorizing structures here. Don't go memorizing, but you should know that purines have two rings. Don't go memorizing where the nitrogens and carbons are located. But just know that one has a 6-membered ring, one's a 5-membered ring. If you see that, you say, well, that's a purine. Now, how do you distinguish adenine purine from guanine? And the answer is you look at the 12 o'clock position. If you see a purine base, two rings, one's 6-membered, one's 5-membered, without worrying about all the nitrogens in the ring and the carbons in the ring, just ask. Look at the 12 o'clock position. If at the 12 o'clock position you see an amino group there, it is adenine. On the other hand, if you do not see an amino group at the 12 o'clock position, it's guanine. One more time. If you see a purine base, you look at the 12 o'clock position. If you see an amino group there, it's adenine. If you don't see it, it's guanine. Now, I'm not trying to bore you with this information, because later on, the last hour I'm here, a week from Friday, we're going to be talking about a major enzyme in metabolism that you already talked about in immunology with Dr. Moscatello, and you'll talk about in a couple weeks with Dr. Barone in pathology. A very important enzyme called adenosine deaminase. Remember the disease adenosine deaminase deficiency? What does the enzyme do biochemically? It deaminates, it removes the amino group from the adenine portion. So again, that's what the enzyme, that's right, it has a lot to do with SCIDs, that's right. But you'll never understand ADA deficiency. You'll never understand the pathology. All the B cell, T cell depletion, you'll never understand it unless you know the biochemistry first. So what does the enzyme do? It removes this amino group from a 12 o'clock position. Now don't worry about the 7 o'clock position. That's not important, the 7 o'clock position. It's what's at the 12 o'clock position of the purine base. Yes, I know some books like First Aid says you can remember these two purines by a little mnemonic or a phrase like pure as gold. What does that do for you? It does nothing for you. Pure as gold. There's too many mnemonics to understand. You know, the First Aid, to understand all those mnemonics in First Aid, you've got to have mnemonics for mnemonics. For the mnemonics, for the more mnemonics. You learn nothing. Just know what I just told you right here. The difference between adenine and guanine. Just pay attention. I'll tell you what you need to know. Now, there are 3 pyrimidines that are important to know, and pyrimidines have one ring. And don't go worrying about what makes up the ring, where the nitrogens and carbons are located. All you need to know with the 3 pyrimidines is the word cut, C-U-T. C is cytosine, U is uracil, T is thymine. Now, how do you distinguish these 3 pyrimidines from each other? Again, you look at the 12 o'clock position. If you see a single ring, you look at the 12 o'clock position. Ask, do you see an amino group at the 12 o'clock position? If the answer is yes, it's cytosine. If on the other hand, there's some sort of deamination that took place, it's uracil. And then how do you distinguish uracil from the other pyrimidine called thymine? And the answer is you look at the pyrimidine, look at the on the periphery of the pyrimidine ring and ask yourself, do you see a methyl group hanging in the breeze? Without worrying where that methyl group is located. Don't worry about where it's located. All I'm saying is, if you see a pyrimidine base with no amino group at the 12 o'clock position, ask yourself, do you see a methyl group hanging, dangling somewhere in the breeze on the pyrimidine base? If there is, it's thymine. Here, don't worry about where it's located. Just say a methyl group on uracil is thymine. And again, I'm not trying to bore you with this information. A few weeks from now, the last discipline you're going to be talking about is pharmacology. When Dr. Harris is here, again, you're going to love Dr. Harris. He's thorough. He's great pharmacologist, our best pharmacologist. When he's here, he's going to be talking about a series of drugs that target this methyl group addition onto uracil. There are so many drugs. I'm going to talk about it too. The last hour I'm here, the same drugs that he's going to be talking about, I'm going to be talking about as well from a biochemical point of view. Drugs like methotrexate, antimicrobial drugs like pyrimethamine, and sulfa drugs, antiparasitic drugs that also target this methyl group on uracil to form thymine. So again, this is major concept. Putting the methyl group on uracil is the target of so many drugs. That's right, Taquir, 5-fluorouracil, many drugs. And you'll never understand the pharmacology, never, unless you understand why. Why are you given those drugs? What are they targeting? The pharmacology always targets biochemistry. And if you know the biochemistry, it goes a long way. All right, now what's coming up next is, so these are the 5 bases that could be found in DNA and RNA. There are other purines and pyrimidines. Don't worry about them now. There are other purines called hypoxanthine. Don't worry about them now. They're not found in DNA and RNA, so don't worry about them. Focus your attention on these 5 right here thus far. Now, of these 5, of the purines, adenine and guanine, both of them are found in DNA and RNA. Cytosine is found both in DNA and RNA. Uracil is found only in RNA. Thymine is found only in DNA. There's a couple of minor exceptions to this. Don't worry about them now. I'll point them out later on. Now, starting with these 5 bases, we're going to start moving up the ladder of complexity to ultimately figure out what is a chromosome. All right, now starting with these 5, what's coming up next are 2 more definitions that we need to attend to. I know it's boring, definitions, but we must attend to the definition. And the first definition to talk about is the definition of nucleoside. You must understand a nucleoside. A nucleoside is one of these 5 bases that you see here. And if we add it onto the sugar ribose or deoxyribose, it's now called a nucleoside. And the next definition is nucleotide. And what a nucleotide is, it's a base, one of those 5, plus the sugar ribose or deoxyribose. And then we add a phosphate, 2 phosphates or 3 phosphates. It's now called a nucleotide. So AMP, ADP, ATP are nucleotides because they have a base, a sugar, and at least 1 phosphate. All right, now what's coming up next is I'm going to show you how a little bit of information in biochemistry goes a long way. Now, in your book, you see on page 6 and 7, you see the structure right here on the left side. Now, suppose on board exams, they gave you this structure, and they say to you, what is this molecule? So you look it over and say to yourself, well, it's not cholesterol. I wonder what it is. So you look it over because you're a thinker. You say to yourself, wow, look, there's a base, a sugar, and 1 phosphate. It's got to be a nucleotide. Now, which nucleotide? It's a nucleotide monophosphate because there's 1 phosphate, some sort of nucleotide monophosphate. Now, then, which one is it? So you look at the base and you say, well, it has a single ring, so it's going to be a pyrimidine. So it's either going to be C, U, or T. And then you look at the 12 o'clock position. Do you see an amino group there? The answer is no. So it's not cytosine. Therefore, it's got to be either uracil or thymine. Now, then you say, well, is it uracil or thymine? Do you see a methyl group hanging in the breeze? The answer is no. So it's not cytosine. It's not thymine. Therefore, it's got to be a derivative of uracil. In fact, it is uracil. Uracil linked to carbon 1' of the ribose and linked to carbon 5'. There is a phosphate. So the answer is it's uridine monophosphate. UMP. By the way, this prime, let me make sure we understand what prime means. Because we got a lot to say about 3', 2', 5', this or that. What that's about is, just to refresh your memory, is there are carbons in the base and there are carbons in the sugar. and distinguish the carbons of the base from the carbons of the sugar, the carbons of the base are just given carbon numbers, like carbon 1, carbon 2, carbon 3, whereas the carbons of the sugar are given prime numbers. So this is carbon 1 prime, carbon 2 prime, 3 prime, 4 prime, 5 prime. So on carbon 5 prime, that means on carbon 5 of the ribose, there is a phosphate. So what the prime numbers are referring to. So this is UMP. Now on the right side, one more practice. On the right side, suppose they give you this structure, even though it says what it is. Suppose they give you that structure and they say, what is this molecule? And again, in my opinion, it's low yield, but you never know. Now, as you look at it, you say, wow, it's a purine because it has 2 rings linked to carbon 1 prime of the ribose, carbon 5 prime, there's a phosphate. Ah, another nucleotide monophosphate. Now, which one is it? Because it's a purine, it's either going to be adenine or guanine. You look at the 12 o'clock position, is there an amino group there? The answer is no. So it's not adenine, it must be guanine. So this is guanine linked to ribose and carbon 5 prime, it's monophosphate. So it is GMP, guanosine monophosphate. Now what does the little d refer to? The little d refers to the ribose. On carbon 2 prime, if there's a hydroxyl group present, that means it's ribose. If on carbon 2 prime there is no hydroxyl group, it's deoxy form of the ribose. It is deoxy form of the ribose, so therefore the product is now called deoxyguanosine monophosphate, abbreviated deoxy GMP. All right, now again, it wasn't hard. I know I'm boring some of you out there, but we have to go through this issue. Now what's coming up next is a major table that you must master. I know it's boring, but you must master the different names. Here are the 5 bases once again. Nothing new, you saw those on the previous figure. And you must know the difference between the base and the nucleoside and nucleotide name. You can't memorize this. You have to think about it and master it. Adenine is the base. If we stick it onto ribose, the product is called adenosine. That's the nucleoside name. If we stick it onto deoxyadenosine, it's called, if we put it onto deoxyribose, it's now called in parentheses deoxyadenosine. I'm not trying to bore you with this. Again, remember the enzyme adenosine deaminase? Think of the name. Look, think of the name. Adenosine deaminase. It's not called adenine deaminase. It's called adenosine deaminase. Why? Because it must see the ribose on the adenine. It doesn't work on adenine. It works on adenine, but the ribose must be present, either ribose or deoxyribose. So adenosine deaminase works on adenosine or deoxyadenosine. You got to know the nucleoside name. It's going to hurt you later on in the last hour I'm here. On page 290, the last chapter, all these stuff. You must know the difference between the base, the nucleoside, and the nucleotide name. If we add 1 phosphate, 2 phosphates, 3 phosphates, you can read the rest of this stuff. Now again, I'm not going to go through this table, but you must know the difference between the base and the nucleoside name. All right, now once the nucleotides are formed, like deoxy-AMP and all these other things, then we could polymerize them to form DNA and RNA. Now what's coming up next is a sequence. There's going to be 2 sequences of nucleic acid. 2 sequences side by side. And so we're going to go through the exercise. Is the sequence nucleic acid on the left side, is it DNA or is it RNA? On the right side, is that DNA or RNA? Okay, because after all, we can have DNA double strand, we can have DNA RNA double strand, or we can have RNA RNA double strand. Remember from Dr. Muscatello's lectures, there are examples of double-stranded RNA viruses. So in nature, we get these different kinds of DNA DNA RNA, or RNA RNA double strands. So the idea is, if you see a sequence of 1 strand, how do you know, what do you look for? Is it DNA or is it RNA? And there are 2 things you look for between RNA and DNA. 2 things. One thing is you look at the pyrimidines. And again, the single rings. And you look at the pyrimidines and ask, are there any pyrimidines that have a methyl group hanging in the breeze? If the answer is yes, that means that pyrimidine base got to be thymine. Thymine's found only in DNA. That's got to be a DNA strand. If you see a methyl group on the pyrimidine base, it must be DNA. Now, on the other hand, the other way to do this, you look at the ribose type. You look at carbon 2 prime. If on carbon 2 prime there is a hydroxyl group, that's ribose, that's RNA. If there is no hydroxyl group, it is deoxyribose, that is DNA. Now I'm going to show you something here because a lot of students forget this. Elementary science. Remember way back in high school when you talked about what's the difference between RNA and DNA. People forget what the letters stand for. The letters RNA stands for ribonucleic acid. It stands for the ribose portion of nucleic acid. Whereas DNA, people forget this. But DNA stands for deoxynucleic acid, and it's the deoxy form of ribose. Remember all that? Probably forgot that. But what RNA means, what DNA means, it's referring to the sugar. There it is. It's referring to the sugar. All right. Okay, now what's coming up next is two strands. Well, I'm looking at the time. Right now it is 9 minutes to the hour. So Beth is going to bring up the 10-minute timer, and then I'm going to go off camera and then be back in 10, and then I'm going to continue the exercise of moving up the ladder of what is nucleic acid and what is DNA and what is a chromosome in this chapter. So let's take a 10-minute break. See you in 10. Welcome back, everybody, for hour two. 44 hours to go. So a lot of stuff coming your way. So you see in this first hour how I do my reviews. I'm driven to make sure that you understand this stuff, and I'm very passionate. Yes, I'm very excited because I want you to know it. I could just be here if I didn't care about you. I would just be saying, purines have two rings, and you can know as pure as gold, and it doesn't do you any good. I'm passionate. You're my people. I want you to know it. I want you to understand. I want you to ace the boards. I want you to do so well. And I'm going to do everything I can to get you to get excited about it too. So yes, I'm overly passionate. Some people say I get too excited because I want you to know it. I want you to do well on boards. There's no greater feeling is when I get an email from a student, a former student, who took the board exams, and they email me and say, Dr. Turco, thank you so much for your input. I did well on my boards. I got a 246 or 250, something like that. And this makes me feel good that in my own little world, I'm contributing to humanity. You know, I want you to do well. So please, do pay attention, and you're going to do well. You're going to do well as long as you stick with it. Don't let anybody else get you discouraged. We as instructors have your back. We know what's important. I know. I've been doing this for 17 years. I got so many emails from students. They tell me what kind of topics are on board. I know what's on the boards. As long as you pay attention, you're going to do fine and understand and not memorize. All right, now what's coming up next is two strands of nucleic acid. Is it DNA on the left or is it RNA on the left? Here it is. So on the left side, suppose they give you this strand here of nucleic acid. Okay, now first of all, what is nucleic acid made up of? DNA and RNA. And the answer is all DNA and RNA, they're made up of nucleotide monophosphates. Base, sugar, phosphate. Base, sugar, phosphate. Base, sugar, phosphate. So DNA and RNA are made up of nucleotide monophosphates joined together. And the name of the bond that joins one nucleotide monophosphate with another is called, what's it called? What's the name of the bond? What's the name of this bond right here that joins two nucleotides together? It's called a phosphodiester bond. Now, where does that name come from without memorizing? Because when the phosphate is linked to carbon 3', it's a monoester linkage there. When the phosphate's linked to carbon 5', it's a monoester linkage there. Mono here, mono there, together. It is a phosphodi, because there are two of them, phosphodiester. That's what it is. The linkage between two nucleotides is a phosphodiester linkage. All right, now is the structure on the left, is it DNA or is it RNA? So you look at the bases, and you say, look for the pyrimidines. Well, two rings, purine, another purine. Here's a pyrimidine. Do you see a methyl group hanging in the breeze? The answer is no. You go to another pyrimidine. Do you see a methyl group? Here's the methyl group. Because there's a methyl group on the pyrimidine, that means it's got to be thymine. It says so. And because it's thymine, it's got to be DNA on the left. And that's confirmed by looking at the 2' carbon. At the 2' carbon of the ribose, there is no hydroxyl group. So it's deoxyribose, confirming the sequence on the left is DNA, one strand of DNA. Now, every strand of DNA or RNA, no matter how long it is, this DNA is 4 nucleotides. But you can have DNA that's thousands of nucleotides in sequence, millions of nucleotides in sequence. And on one end, generally speaking, there'll be a 5' phosphate. It doesn't have to be, but for the boards, usually there'll be a 5' phosphate, and on the other end, there'll be a 3' hydroxyl, no matter how long it is. It doesn't have to be, but generally there's a 5' phosphate on one end and a 3' hydroxyl on the other. All right, now the sequence on the right, is it DNA or is it RNA? Because after all, this could be DNA double strand. This could be DNA RNA hybrid. Okay, so on the right side, so we look for the pyrimidines. Here's a pyrimidine. Do you see a methyl group? The answer is no. Here's another pyrimidine. Do you see a methyl group? Here it is, the methyl group. Because it's a methyl group on pyrimidine, it's got to be thymine. And because thymine is found only in DNA, this has got to be another strand of DNA. And that's confirmed by looking at the 2' carbon. There is no hydroxyl group. So this is a DNA double strand. Easy to figure out. DNA double strand. But notice here, importantly, on the right side strand, here is the 5' phosphate, and here is the 3' hydroxyl. In other words, these two strands look like they're going in opposite directions. And in fact, that is one of the two double-stranded DNA properties, DS, double-stranded DNA properties. Also could be double-stranded RNA properties or double-stranded DNA RNA properties. And there are two properties you must know. Again, this is elementary molecular biology. These are anti-parallel strands. These strands always go in the opposite directions. So the 5' phosphates on one end to the 3' hydroxyl. On the other strand, it's just the reverse. On one end, here's the 5' phosphate, all the way to the 3' hydroxyl. So in DNA and RNA, there are anti-parallel strands. No exceptions in life. Human cells, microbes, yeast cells, plant cells, anti-parallel strands. Now, the other important feature is that what holds these two strands together? And what holds these two strands together is these hydrogen bonds. These are hydrogen bonds where G base pairs with C, A base pairs with T. This is now the other property that you must know. These bases are complementary. G always base pairs with C, A always base pairs with T. Now, if one or both strands are RNA, there's no T in RNA. So A base pairs with U. So one more time. G always base pairs with C, whether it's DNA or RNA. And if there's DNA strands, A base pairs with T. But if one of the strands or both is RNA, A base pairs with U, because there's no T in RNA. Always, always, always. No exceptions. That's normal. Be ready for pathology coming up tonight. Be ready for it. All right, now another item here. Notice here, don't worry that there's 3 hydrogen bonds with C and 2 hydrogen bonds with A with T. That's low yield. Don't worry about that. But the idea is what can separate these two strands? Let me see your answers here. What can separate the two strands? What separates, so what's holding together are these hydrogen bonds. That's right. Heating, melting, that's right. So in a test tube, what separates the strands from each other is heating. If I take double-stranded DNA and heat it to 50 degrees, 60 degrees, the two strands will come apart. That will be important in Chapter 7. Right now we're in Chapter 1, but in Chapter 7, we're going to be talking about a very important technique called the PCR reaction, polymerase chain reaction. And in the PCR reaction, we need to use heat to unwind DNA. All right, now there's another item here, basic molecular biology, the way you name nucleic acids like DNA. So on a board exams, what they might do is they might show you a sequence, one strand of DNA that looks like this. They actually show you the structure. Or they might spell out the structure and say 5' TCAG to the 3' end. So the sequence of DNA actually is this sequence shown on the left. 5' TCAG to the 3' end. So these letters, these are not bases. Please keep in mind these are not bases. These are nucleotide monophosphates, specifically deoxy form. So it's 5' TCAG to the 3' end. Now another way you might see this on board exams, you might see it without the numbers. You might see 5' TCAG. Now let me stress, get ready with your highlighter. On board exams, if they don't show you the orientation, if they don't say which side is the 5' or the 3', you must, did I emphasize it enough yet? you must assume it's the 5' end on your left. If they don't show you the numbers, you must assume it's the 5' end on the left, unless they say otherwise. The only way it would be the 3' end on your left, you must see the number. So you always read DNA and RNA 5' to 3'. The only way it would be the 3' end on your left, you must see the number on a computer monitor when you take the board exams. You must see the 3'. If you don't see it, you must assume it's the 5' end on your left if you don't see the numbers. Now, if you're thinking, what's the big deal? So what? Trust me. A little bit later, I'm going to ask you a question. And many of you in the audience right now are going to get it wrong. Why? You forget what I just told you. Trust me. How do I know this? History tells me so. Every time I give that question, students miss it. Oh, yeah, he did talk about this. Trust me, it's going to come later on. A lot of you are going to forget that. Why? Because some of you are texting your buddies and so on. Be ready for what I'm emphasizing what you need to know. All right, now what's coming up next is, now moving up the ladder of complexity, what have we done? We talked about what a base is, what a nucleotide, what one strand, two strands are all about, what joins them together, these hydrogen bonds. Now, we're going to skip over a couple of low-yield items, and that's the issue of the double helix. You know, nowadays they talk about the double helix in high school. But the double helix is where you twist the DNA, and then you get supercoils where the DNA gets over tightened. We're going to skip over all that stuff. Low yield. There is one very important enzyme with supercoiling that we'll talk about in chapter 2. Don't worry about it in chapter 1. But the idea is the double-stranded DNA can twist to form the double helix. It can over tighten by supercoiling. And now we're going to move up the ladder of complexity even more and talk about what a portion of a chromosome actually looks like. Now, this is going to be extremely high yield. The integration of biochemistry with histology and cell biology. So the idea is, in the blue, this wavy part right here represents double-stranded DNA. And a portion of the chromosome, the double-stranded DNA can wrap around protein. So if I take one of these little discs, so if I take a disc from a chromosome and magnify it, this is what it looks like. So in the blue, this represents double-stranded DNA that wraps around protein. It wraps around the protein twice. And importantly, this disc in cell biology and histology is called a nucleosome. And when a nucleosome is equal to DNA wrapped on the outside of proteins. The class of proteins are called the histones, and histones are positively charged. And why are they positively charged? Because of the amino acids that are in them. Don't worry about them now. We'll talk about the amino acids later on. But these are dominated by two kinds of amino acids, lysine and arginine. Dominate histones because they are positively charged. And they bind to the negatively charged DNA. Let me see your answer here. Let me see your answers. Why is DNA negatively charged? What makes DNA negatively charged? It sounds obvious, but I'm making sure you understand. Why is DNA negatively charged? Not the amino group. It is the phosphodiester. Remember the phosphodiester? Each phosphodieester is one negative charge. So if you have hundreds or thousands of phosphodiester bonds in a big DNA strand, it's highly negatively charged. Please keep that in mind. That's going to be important later on in chapter 7 when we talk about the southern blot, how you electrophoresis DNA, separate DNA. It's all coming up a little bit later, but it's based on the charge here. Okay, the DNA is negatively charged. So the histones on the interior bind, they're positively charged, they bind like a magnet, they bind to the negatively charged DNA. Also high yield is the class of histones and the examples of histones, and there are 5 of them you must know. And they are H1, H2A, H2B, H3, and H4. These are the 5 histones that you must know. Now, of these 5 histones, it's the bottom 4, H2A, H2B, H3, and H4 is where you have two copies of them that make up a nucleosome. So in the interior, in this diagram here, remember it's 2 dimensions, but in reality it's 3 dimensions. What you're looking at is one copy of H2A, H2B, H3, and H4. The other copy faces the inside that you can't see. So let me stress, a nucleosome is an octamer. Octamer. 8 proteins make up a nucleosome because there's 2 copies of H2A, H2B, H3, and H4 here. So it is an octamer, is a nucleosome. On board exams, they might call it a nucleosome, these little discs, but they might also call it the 10 nanometer chromatin. So if you see that phrase, 10 nanometer fiber, but more commonly 10 nanometer chromatin, you say, oh, they're asking me something about the nucleosome. Okay, that's the diameter of the disc, 10 nanometers in diameter. Now what about H1? Notice here it is a histone. It's shown here in the off color, the gray color, just to show that it's not in a nucleosome. H1 is found outside the nucleosome. And what H1 does, it condenses. It's condensing the nucleosomes much more closely together. So when H1 is present, the nucleosomes are much more condensed together. And there the diameter of all these discs together is now called the 30 nanometer chromatin. So here it is. If on board exams they ask you, what's the difference between the 10 nanometer and 30 nanometer chromatin, you answer the presence or absence of H1. When H1 is present, the nucleosomes are much more condensed together, compacted together. Without H1, they are much more loosely associated with each other. So the presence or absence of H1. Okay, now what's coming up next? The last figure in this chapter, and then we're done with chapter 1, is very high yield. More integration of biochemistry with histology and pathology and cell biology, but also a lot of concepts that students, with the stuff coming up in the next figure, some students never thought of this. It never dawned on them. Oh yeah, now that makes sense. Trust me, some of you never thought of this stuff coming up on the next figure. And that is, what is a chromosome? Yeah, it's made up of DNA. Yeah, it's made up of protein. But what is it? Okay, that's coming up on the next figure. Major figure. A lot of concepts coming up on the next figure. I told you I'm a big picture type of guy. Get ready for it on the next figure. All right. Now, at the top of the figure, in this high yield figure, is we talked about earlier, if we take 2 strands of DNA, and then we twist it, that forms the double helix. So this is now naked DNA, 2 strands of DNA wrapped, forming twisting the double helix. And then when we add 2 copies of H2A, H2B, H3, and H4, that forms a 10 nanometer chromatin. These also called the nucleosomes. But notice they're still loosely associated with each other, just loosely associated with each other. But then when we add H1, when H1 is around, we get much more condensing that goes on. We form the 30 nanometer chromatin, nothing new. And then when other proteins are added, and these are sometimes called the scaffolding proteins, it does even more condensation, much more compacting together when these scaffolding proteins are present. And that's all called euchromatin. So all the stuff here on the left is called euchromatin. But then when other proteins are added, now the name of these other proteins have never been on board exams. Don't worry about them. Low yield. But when these other proteins are added, then we get the highest order packing of the chromosome. And that's, in cell biology, is called heterochromatin. One more time, heterochromatin is the highest order packing of the chromosome. You can't even see the DNA. The DNA is surrounded by protein. The DNA is, none of the DNA is exposed. That's called the heterochromatin. So euchromatin is all the stuff here on the left, and on the right side is the highest order packing when these other proteins that are not important for boards are added, called heterochromatin. Now here's a concept that a lot of students never thought about. If I took any of your chromosomes, remember you have 46 chromosomes now in your cells. 23 came from your mother, and 23 came from your father. If I took any of the chromosomes, any one, and just like and scanned it from top to bottom. You've seen all those scanners, you know, those red line scanners. You've seen those things, how they scan something, you know. If I were to do the same thing, just visually, just imagine now. The idea is if I took a chromosome and just made it vertically and put the scanner at the top and scanned it from top to bottom. I scanned the chromosome from top to bottom. Depending on where I'm at on that chromosome, a portion of the chromosome might look like this. A portion of the same chromosome might look like this. A portion of the same chromosome might look like this, where the green pointer is. A portion of the same chromosome might look like this. And a portion of the same chromosome might look like this with heterochromatin. And it all depends where we're at on that particular chromosome. What that chromosome looks like, where we're at on that particular chromosome. So in one chromosome, we may have heterochromatin domains, and we may have euchromatin domains. And these euchromatins might look like these various pictures like you see right here. All right, now, again, people never thought about that, but that's what a chromosome is. Portions of it are heterochromatin, some of it is euchromatin. All 46 chromosomes look like that. All right, now in cell biology and histology, euchromatin is sometimes called the light staining spots of a nucleus, whereas heterochromatin is sometimes called the dark staining spots of a nucleus. Now, what I mean by this is, in the cell biology histology, on page 12, you see this electron micrograph. Now here comes, what I'm going to do is, I'm going to talk about this electron micrograph, and I'm going to ask you a series of questions. Get ready with your fingers. Let your fingers do the talking. Now again, if you looked at the online demand videos and you got the answer in front of you, please don't answer, because give everybody a chance here. So please don't answer if you have the answer in front of you. Don't answer. But the idea is, suppose they say to you on board, suppose you're taking the board exam, and up on the monitor comes this electron micrograph. And suppose they say to you, this is an electron micrograph of a nucleus, of a muscle cell, of a female. That's the information they give you. All right, here comes a question for you. Now, if they have arrows pointing to the various parts of the nucleus, they might ask you straightforward cell biology, what is it? So if they have an arrow pointing to the light staining spots of a nucleus, the light staining spots, and ask you, what is it? You say, well, that's euchromatin. If they have an arrow pointing to the multitude of dark staining spots of a nucleus, the multitude of dark staining spots of a nucleus, and ask you, what is it? You say, well, that's heterochromatin, because heterochromatin is the dark staining spots of a nucleus. If they have an arrow pointing to the single biggest spot of the nucleus and ask you, what is it? Identify it. And you say, that's the nucleolus. Now, suppose they do it and they make the question a little harder. Suppose they say to you, this is a nucleus of a muscle cell of a female, and they have an arrow pointing to the light staining spots of a nucleus, and they don't ask you to identify it as euchromatin. They're asking you, what's happening there? Let me see your answers. What's happening in the light staining spots of a nucleus? What's happening in the light staining spots of a nucleus? And the answer is, it is active gene expression, transcription. You're making RNA because the DNA is exposed. Look how the DNA is exposed in euchromatin. When DNA is exposed, RNA polymerase coming up in chapter 3 can turn on the gene and make RNA from that particular gene. Now, suppose they have an arrow pointing to the multitude of dark staining spots of a nucleus. They're not asking you to identify it as heterochromatin. They're asking you what's happening there. Let me see your answers. What's happening in the dark staining spots of a nucleus? Nothing's not, I need more detail, Sagar. I need more detail. Talk with her. I need more detail. Vincent, nothing. Nothing doesn't mean anything. Nothing means nothing. What is happening there? And your answer is, there we go, Sagar, there we go. Now we're talking. That's where you have inactive genes. Inactive genes is in the dark staining spots of the nucleus. All right, another question. Now, suppose they have an arrow pointing to the dark stains. Now, again, if you have the answer in front of you, please don't respond. I'm trying to get people to think, to try to recall things from other disciplines. Suppose they have an arrow pointing to the multitude of dark staining spots of a nucleus. And they're not asking you to identify it as heterochromatin, because it is. They're asking you what's happening there. And suppose one of the 5 options is not inactive genes. Let me see your answer. What else is happening in the dark staining spots of a nucleus? Give everybody a chance to respond. Don't go looking at other people's responses first. Type in your response. That's right, Sanan, Tariq, and there it is. And you answer by saying that's where you have bar bodies. Notice female. Because in the chromosome here, in the chromosomes, remember the female is going to have 2 X chromosomes. And one of the X chromosomes is entirely heterochromatin. One of the X chromosomes in a female, the cells of a female, is entirely heterochromatin. It looks like this. The entire bar body, it looks like this. But the other X chromosome that she has, the other X chromosome in this particular nucleus, a portion of that X chromosome looks like this, a portion of that X chromosome looks like this, a portion of the X chromosome looks like this, a portion of the same X chromosome looks like this, or looks like that. In other words, bigger picture. In a female, remember the gene G6PD. Remember that major enzyme? I got a lot to say about that enzyme later on. Glucose 6-phosphate dehydrogenase that you hear about in all courses. That gene is located on the X chromosome. And in the nucleus of a female, one of the G6PD genes is in the bar body. It's in heterochromatin, never to be turned on. The other gene for G6PD is in euchromatin, to be turned on. Again, the concept, part of the big picture. But now, one more item here. If they say to you, the nucleus of a muscle cell of a male, you don't say bar body, because the male only has 1 X chromosome. And that 1 X chromosome, it could be euchromatin portions or heterochromatin portions. Again, a lot of people never thought about that, but you got to know what's going on here. Now, another item here, another question. Get ready with your fingers. Suppose they have an arrow pointing to the light staining spots of the nucleus, and they're not asking you, what is it? You would say that's euchromatin. And suppose they ask you what's happening there, and suppose one of the 5 options is not active genes. Let me see your answers here. What else is happening in the light staining spots of a nucleus? Not transcription is active genes. Now think, replication. Angela, think. Hector, replication, Hector. Now think. See, this is why I'm glad you're responding. Even though you gave the wrong answer, you'll never forget the right answer. Notice here, one word, muscle. Remember, muscle cells are in G0 phase. Muscle cells do not undergo replication. And so as a result, it's not replication. What happens there is let me see the answer here. You could say translation, that's true, but the answer is apoptosis. When the DNA is exposed, DNA is susceptible to nuclease degradation, and that's what can go on in the light staining spots of a nucleus here. So please be ready for that. That's how they ding you on boards if you're not paying attention to the words. Muscle cell, that's cells in G0 phase. We talked about that in the first hour, cells in G0 phase. High yield, you got to know what cells in the body are in G1 or G0 phase. And female versus male. All right, look at the concepts here, part of the big picture. All right, another question for you. Now in chapter 6, later on in chapter 6, we're going to talk about cloning. how you clone genes and what are you going to do with a clone gene. Cloning means you make millions of copies of a particular gene. Now suppose I want to clone the Turco insulin gene. I want to make millions and millions of copies of the Turco insulin gene. And to make millions of copies of it, I have to get it somewhere in my body to start the cloning process. And so the idea is, where would I get the insulin gene from Turco to start the cloning process? So here comes the question for you. Where would the insulin gene be in the beta cell? So in other words, if I do a biopsy of my pancreas and take out the beta cell, because everybody knows the beta cell secretes insulin. So if I take out the beta cell, where would the insulin gene be located in the beta cell? Would it be located in euchromatin, heterochromatin, or not present? Very simple question. Is insulin gene in the beta cell? Is in euchromatin, heterochromatin, or not present? Everybody should be saying it's in euchromatin, because everybody knows insulin is a protein. And because it's a protein, it comes from RNA. RNA comes from DNA. And DNA has to be transcribed. You got to know what that word means, transcription, making RNA. You got to master that word. So the gene for insulin has to be transcribed into RNA, RNA synthesis. Then RNA is made into the insulin protein. Everybody knows this. There isn't one student listening to me right now that doesn't know that the beta cell secretes insulin. So you've got to know that the insulin gene is in euchromatin in the beta cell. All right, that's a simple question. Now, slightly harder question, slightly harder. Where would the insulin, now if you're thinking G0 phase and all that, that has to do with replication. We're talking about transcription. Don't get those words confused, replication and transcription. I'm telling you right now, you must master the term. Tonight when we're done, go back until you know it inside and out what replication means, transcription, translation. You're going to be bogged down, you're going to be hurting over the next couple of days if you don't know what the words mean. All right, comes another question. Suppose I do a biopsy of my pancreas and I don't get the beta cells, I get the alpha cells. Where would the insulin gene be in the alpha cell of my pancreas? Would it be in euchromatin, heterochromatin, or not present? Okay. Think. Would it be okay? All right. And now a lot of people think it's not present. It is present. It is in heterochromatin. The insulin gene is in every cell in the body. Every cell is I could do a brain biopsy and there's the insulin gene. But in all the cells in the body except the beta cell, insulin gene is in heterochromatin. Just like here, see the green arrow? Insulin gene is covered up. You never see it in all the other cells in the body. The only place the insulin gene is in euchromatin, it's in the beta cell. But the insulin gene is in every cell in the body. Now the why question, why? Why is that? And the reason is think and you'll get the answer. By the way, let me ask you this. How many genes, you have 46 chromosomes. 23 chromosomes came from your mother and 23 chromosomes came from your father. Let me ask you this. The chromosomes that you got from each parent, from your mother, for example, how many genes are in 23 chromosomes? Let me see here. Roughly speaking, how many genes are in 23 chromosomes? Not a million. No, much less. The answer is, here comes the answer, there's only about 20,000 genes that you inherited. They're in 23 chromosomes that you got from your mother, and the same 20,000 genes and 23 chromosomes that you got from your father. So you inherited an insulin gene from your mother and an insulin gene from your father. Remember, we are diploid. We have 2 copies of all the genes here, except for the genes on the Y chromosome. But don't worry about that right now. Just think concepts. The idea is there's roughly 20,000 genes. Remember, the human chromosomes have been sequenced. And the latest estimate is we have about 20,000 genes. Not much. You think it'd be a lot more. You think millions. It's not. So the idea is you have roughly 20,000 genes in 23 chromosomes. and that you got from your mother, and the same 23 chromosomes, roughly speaking, except for the Y chromosome, that you got from your father here. Okay, but now the issue is, here comes the question, here comes the point. But remember, in fertilization of the sperm and the egg, in the sperm, there are 23 chromosomes with 20,000 genes, including the insulin gene from your father. The egg of your mother had 23 chromosomes and the insulin gene in those 23 chromosomes somewhere. And in fertilization, we form the zygote. And now we have 2 copies of each gene. And then we get the embryonic stem cells, and eventually the embryonic stem cells differentiate into pancreatic cells, muscle cells, brain cells, hepatocytes, and so on. And during that differentiation, all 20,000 genes times 2 are present in every cell in the body, because you were a zygote with 46 chromosomes, then you became 2 cells, 4 cells, 8 cells, 16, 32, 64, 160, 64, 128, whatever, I don't know, all the way to 10 trillion cells. And the insulin genes is in every cell in the body times 2. You have 2 copies of them, except the erythrocytes, because erythrocytes don't have a nucleus. But just picture, in my brain, I could do a biopsy of my brain and put it in a test tube. And if I get rid of all the proteins, see all these proteins here? They're making heterochromatin and so on. If I get rid of all the proteins with detergents, I got negative DNA in that test tube. And in that negative DNA are 20,000 genes in 23 chromosomes from my mother, and 20,000 genes and 23 chromosomes that came from my father. Some students never thought of that, but that's life. That's life. Okay, everybody understand what goes on? So if I'm going to clone the insulin gene, I can get it from any cell in my body. Put it in a test tube, get rid of the histones. By the way, when I say get rid of the histones and the proteins, don't worry about the details. Let PhDs worry about that. It's just easy to do. You add some detergent in a test tube, all the histones go away, and you got negative DNA, chromosomal DNA from 46 chromosomes. And somewhere is 2 copies of the insulin gene. There it is. Concepts. Concepts. Don't go memorizing. Just think about it. Oh, yeah. It makes sense. Right? Yeah, it makes sense. Okay. All right, everybody. We're finally done with Chapter 1. Okay? The high-yield items in Chapter 1 covered. As long as you were paying attention, you know what's important and what's not important in Chapter 1. All right. Now, what's coming up next is Chapter 2. And in Chapter 2, we're going to talk about the basics of replication, DNA replication, what you need to know and what you don't need to know. And trust me, there's a lot of stuff you don't need to know, low-yield stuff. As long as you're paying attention, I got your back. But if you're studying on your own, you got your work cut out for you, because there's a lot of garbage here that you're wasting time on. I'll tell you what you need to know about the basics of replication. And then after that, we're going to talk about DNA repair mechanisms, high-yield integration with pathology that Dr. Barone will also talk about a little bit later. All right, now the basics of replication. Now what's coming up next on page 19 is a high-yield figure in terms of concepts. Concepts here. Big picture item here. All right, now what we're doing here is a big comparison of what you need to know about how a DNA polymerase works compared to how an RNA polymerase works. On the left side is how a DNA polymerase works, what you need to know in terms of the basics. And on the right side is how an RNA polymerase works. There's some very important similarities and some major differences between these 2 polymerase enzymes. All right, now we'll focus our attention on the left side. How does a DNA polymerase work? Now, the way I'm going to approach this is I'm going to give you 5 numbers. Each number, I'm going to talk about it, and then after we talk about DNA polymerase, the same 5 numbers, I'm going to compare how the RNA polymerase works. There's very important similarities and some very important differences. All right, now suppose here in the blue rectangle with the letters, that represents one of the 2 strands of double-stranded DNA. So the double-stranded DNA of the chromosomes, again, they can open up, and what we're looking at, focus our attention on one of the 2 strands. And it says on your left side is the 3' end, And it says on the right side is the 5' end of the strand of DNA. And the circle represents DNA polymerase. Okay, question number 1. If the National Board asks you, how does the DNA polymerase know where to sit down on the DNA to begin the process of replication? The DNA polymerase does not sit on the DNA randomly. It sits, and you answer by saying what a DNA polymerase has to see. It has to see an RNA primer. So an RNA primer is laid down first. And notice how the RNA primer is shown here in the purple with the white letters, how the RNA primer is complementary, A with the T, C with the G, U because it's RNA with the A, G base pairing with the C, and anti-parallel. DNA is 3' to 5', the RNA 5' to 3'. Always, always, always. So the RNA is base pairing, complementary, anti-parallel fashion. So the primer, the RNA primer, serves as the chair to which the DNA polymerase sits down. Item number 2, which direction will the DNA polymerase go? Conceivably, it can go from your right to your left, or from your left to your right. Which direction will it go? And the answer is right here. The answer is all DNA polymerases read the template 3' to 5'. There is no exceptions to that in life. Microbes, eukaryotic cells always read 3' to 5'. So in this example, it's reading from your left to your right. Item number 3. So the DNA polymerase, when it sits down on the RNA, it's going to read the template. It sees a deoxy TMP in the DNA, and it puts in deoxy AMP in the newly synthesized DNA. Here's a deoxy AMP and makes it into deoxy TMP. Here's a deoxy GMP in the DNA template, puts it in a deoxy CMP in the newly synthesized DNA. Notice how the DNA shown in the white rectangle with the black letters, notice how the DNA that's being made is being made in a 5' to 3' direction. Always, always, always. By adding nucleotides to the 3' end, DNA synthesis is always 5' to 3'. No exceptions in life. Item number 4. So then what can happen is the DNA polymerase can make a mistake. Here is a deoxy GMP in the template. In the newly synthesized DNA, this should be deoxy CMP. But the DNA polymerase makes a mistake. It puts in a deoxy TMP. Before we get to that mistake business, I forgot to mention one thing. That even though DNA is made up of deoxynucleotide monophosphates, the enzyme does not polymerize deoxynucleotide monophosphates together like Legos. You know what Legos are. The enzyme does not polymerize deoxynucleotide monophosphates together like Legos. What happens is the enzyme needs all 4 deoxynucleotide triphosphates. It needs deoxy ATP, deoxy TTP, deoxy CTP, and deoxy GTP. And because the high energy triphosphates are needed, monophosphates are added, you got to ask, what happens to the other 2 phosphates? They come off as inorganic pyrophosphate. So PPI, inorganic pyrophosphate. So 2 phosphates are removed from the nucleotide triphosphates. Not now. I'm going to ask you why. Why doesn't the enzyme DNA polymerase polymerize like Legos? You know what Legos are. Why doesn't it polymerize like Legos deoxynucleotide monophosphates? Why do you need the high energy triphosphates? The answer comes up later. Don't worry about it now. But triphosphates are needed. Okay, but now coming back to item number 5. So in this example over here, there's a deoxy GMP in the template. This should be deoxy CMP in the newly synthesized DNA. Clearly a mistake. It's not paired properly. Fortunately for me and you, our DNA polymerase pauses and checks what it has done. When it realizes, oh man, I made a mistake. I made a mistake. Fortunately for me and you, within DNA polymerase, it has 3' exonuclease activity. 3' exonuclease activity. On that 3' end, see right there's a 3' end. On that 3' end of the DNA, it takes out the entire nucleotide. In this example, deoxy-TMP. So it takes it out because it proofreads, the enzyme DNA polymerase proofreads, and because it has 3' exonuclease activity, it takes out the mispaired nucleotide. And now notice how it's vacated, right here, it's vacated because it takes out, in this example, deoxy-TMP. And then before DNA polymerase continues, it puts in the correct nucleotide. There it is, there's the correct nucleotide, deoxy-CMP, and then DNA polymerase continues to go on. And this is what's referred to as high fidelity DNA synthesis. Now in a few minutes, I'm going to ask you why. Why is our DNA polymerase high fidelity? That's coming up in a few minutes. All right, now on the right side, suppose now this is some template sequence, and this might be some gene. And here is the enzyme RNA polymerase in the red circle. So item number 1, if you were asked how does a DNA polymerase know to sit down on the DNA to begin the process of replication, you say an RNA primer. But notice here, item number 1 with RNA polymerase, a primer is not required. Uh-oh, a primer is not required. Because, now think about it, then you should ask, how does the RNA polymerase then know where to sit down on the DNA? Much more importantly, how does the RNA polymerase know where a gene is located? Now think, here comes part of the big picture. Part of the big picture of life, integration with physiology. What I mean by this is, the break is coming up another couple minutes here. When the break comes, a lot of you are going to go, you might go and eat a donut. And when you eat the donut or a piece of chocolate, what's going to happen? Your blood glucose is going to rise. The biochemistry of that we'll talk about later on. But because you eat all that donut or you eat a piece of chocolate, your blood glucose will rise. And because the blood glucose rises, you're going to secrete insulin. And then insulin will go to certain tissues of the body. And when insulin knocks on the door of those tissues, insulin will bind to its receptor and then signal the RNA polymerase of that cell. Insulin basically is telling the RNA polymerase, go into the chromosomes and find a key genes in the pathway of fatty acid synthesis. And sure enough, RNA polymerase will obey the insulin. RNA polymerase will find the key genes for fatty acid synthesis. So as a result of eating that donut, you're going to take the carbohydrate donut in the donut and make fatty acids. You know this. If I ate 5 donuts right now, I'm going to gain weight. The biochemistry of the physiology here. How does insulin then signal RNA polymerase to turn on gene expression? How does one enzyme, think about this, this is staggering. How does one enzyme, RNA polymerase, know in 46 chromosomes where the genes for fatty acid synthesis are located? I mean, think about it. How does one enzyme, RNA polymerase, knows where the genes for fatty acid synthesis are located and turn on those genes and not the other 20,000 genes? That's staggering if you think about it. One enzyme, RNA polymerase, under the command of insulin, knows where the genes are located. Maybe you didn't know this. Maybe you didn't know this. But your genes don't have neon signs. It's not like you're in Times Square. I was in Times Square yesterday. I just flew back from New York yesterday. I was in Times Square, all these neon signs, all the lights and so on. Your genes don't have that. You know, think about it. Your chromosomal DNA are 4 nucleotides in some sequence. How does one enzyme know where the genes for fatty acid synthesis are located? What chromosome number they're on? And turn on those genes and not the other ones. Not only that, tonight, when you're sleeping in bed, and you're thinking, you're daydreaming about all this biochemistry that's starting to make sense. You're thinking there, because you haven't eaten anything. Your blood glucose is dropping. You're not secreting insulin anymore. But what are you secreting? You're secreting some hormone called glucagon because the blood glucose is dropping. And glucagon is going to go to your liver and tell the liver, hey liver, go tell your RNA polymerase to go into the chromosomes and find a key gene in the pathway of gluconeogenesis, the genesis of glucose. And sure enough, under the command of glucagon, RNA polymerase will find the key gene for gluconeogenesis and turn on that gene, and as a result that you make RNA, you'll make protein, and you'll turn on the pathway of gluconeogenesis. Why? Because glucagon in endocrinology was around. Think about that. That's staggering. How does the RNA polymerase know where the genes are located and turn on those genes and not the other 20,000 genes? It's staggering molecular biology. Now, if you don't know the answer, stay tuned. That's coming up later in chapter 3, but much more detail in chapter 5. Regulation of gene expression, how you turn on genes. The National Board loves that molecular endocrinology. The integration of all those hormones, how do they work? How do they turn on genes, whether it's testosterone or whatever? How do they turn on genes with RNA polymerase? They love that topic for boards. You must master that. All right, now what we're going to do is, I notice the time, it's 7 minutes to the hour. We're going to take a 10-minute break, and I'm going to come back and continue on with RNA polymerase. But all I'm doing is setting the stage for the much more interesting things that you should want to know, not memorize. You should want to master. Here, all right, let's take a 10-minute break. Welcome back, everybody, for hour 3. We're rolling right along, right where I want to be. And so we're talking about this all-important enzyme called RNA polymerase. And magically, it finds genes. It knows where genes are in your 46 chromosomes. So you can imagine there's going to be something really going to be interesting coming along. And the national board hammers medical students on these things that we talked about later on. So don't worry about it now. Don't worry about what does an RNA polymerase have to see on a gene to sit down to begin the process of transcription. That's all coming up later on. There's only so much I can talk about at one time. But a primer is not required. Now, assuming RNA polymerase knows where to find the gene, item number 2. RNA polymerase sits down, which direction will it go? It can go from right to left or left to right. Now, the item number 2 is similar, identical with the DNA polymerase because RNA polymerase reads the gene 3' to 5'. Always, always, no exceptions. So in this example, it's going to go from left to right. Item number 3. So when RNA polymerase starts reading the gene, it sees a deoxy TMP in the gene, makes it into 5' AMP in the RNA. Here's a deoxy AMP in the gene, it puts it into UMP in the RNA. And then there's a deoxy GMP in the gene, puts it into deoxy CMP. So notice how the RNA that's being made is being made 5' to 3' direction, identical with DNA polymerase. So item number 3. DNA polymerase reads 3' to 5' and makes DNA 5' to 3'. Same thing with RNA polymerase. It reads the gene 3' to 5' and makes RNA 5' to 3'. Item number 4. If RNA is made up of deoxynucleotide monophosphates, then the thing is, please pay attention, by the way, in the public chat pod, please pay attention. Don't go make another. I mean, you're not paying attention if you might be missing something that I'm going to say is high yield. Please pay attention. I don't want to waste your time here. Pay attention. Don't go worrying about other people. Be concerned about mastering the material for yourself. Then you can help other people out. But you must understand the material first, please. All right. Now, item number 4. If RNA is made up of nucleotide monophosphates, just like DNA polymerase, what are the substrates for the RNA polymerase? And it's the same thing with DNA polymerase. It needs all 4 nucleotide triphosphates, ATP, UTP, CTP, and GTP. And because triphosphates are needed, monophosphates are being added, you got to ask, well, what happened to the other 2 phosphates? They come off as inorganic pyrophosphates. So 2 phosphates are removed in order to make RNA. And later, not now, I'm going to ask you the why. Why do you need high energy to make RNA? Why you need high energy precursors to make RNA? Why you need ATP, CTP, UTP, and GTP? All coming up. You can memorize, but unless you understand why, you're just going through the motions. Now, item number 5. The RNA polymerase can make a mistake. Exactly what you see here. The gene has a deoxy GMP. This should be CMP in the RNA being made, clearly mispaired. So as a result, here the RNA polymerase, unlike the DNA polymerase, does not pause and check. So the mispaired nucleotide is not removed, unlike the DNA polymerase, which does remove the mispaired nucleotide. And so the RNA has the U at this position, and notice there's an incorrect nucleotide in the RNA being made. And then RNA polymerase finishes off. This is what's referred to as low fidelity RNA synthesis. Whereas DNA polymerase is high fidelity, RNA polymerase is low fidelity. Here comes the why. Why is that? Now you got to think now. Why do you think the DNA polymerase must be high fidelity? If you think about it, it makes sense. Why must the DNA polymerase be high fidelity? Sorry about that. My wife will get the phone. You got it there. Sorry about that. But life. Life is not perfect. I told my wife, get the phone, get the phone. You know. Anyway, gotta be patient. You gotta be patient. But the spouses, you know. All right. So, any case, now the question is, why is it that DNA polymerase has to be high fidelity? Let me see your answers here. Why must it be to avoid mutations in DNA? Gabriel, you're on the right track, but you're on the right track. Mutations. What do you mean by that? Though? I need more, little bit more detail. You're on the right track. You're on the right track. And that's right, because it could be permanent if it's not corrected. In other words, suppose this blue circle represents DNA polymerase in somebody's germ line. And whether it's male or female, making eggs or sperm. But the idea is when DNA polymerase is replicating somebody's DNA in the germ line, if that mutation, if that that incorrect nucleotide is not corrected like it should, if it stays incorrect, then the DNA, that gene, will have a mutation in it that can then be passed on to a progeny. And now remember, 3 nucleotides make up a codon. A codon makes up and encodes for an amino acid of a protein. So that incorrect nucleotide will give rise to an incorrect codon, incorrect amino acid of a protein being made. Now notice the words high fidelity. It doesn't say perfect fidelity. If it was perfect, we would never have disease. But it's high fidelity, not perfect fidelity. Now, what I mean by that is, can you think in pathology? What's the best example of a disease where somewhere in the history of humankind, a DNA polymerase in somebody's germline, whether male or female, when they got to a certain gene, there was a mutation, incorrect nucleotide that was not corrected and was inherited from that into that person's progeny, and then passed on from generation to generation. What's the best example of this? The best example of this, Angelman, yeah, that's true. The best example is Gabriel Sima. That's right. It is sickle cell anemia. Somewhere in the history of humankind, whether it's 1,000 years ago or 2,000 years ago or wherever, somewhere in the history of humankind, in some individual, in the germline of that individual, when a DNA polymerase in that person's germline was replicating that person's chromosomal DNA, when that DNA polymerase got to the beta globin gene at position 6, codon 6, there was an A was made into a T, and it was not corrected. The T remained, the oxy-TMP remained in the beta globin gene. As a result, that was then passed on to that person's progeny, and then the beta globin gene has a mutation in it, giving rise to a defective beta globin protein. As a result, the beta globin protein doesn't fold properly, hemoglobin doesn't fold properly, red blood cells doesn't work properly, giving rise to sickle cell anemia. All because one little defect in biochemistry. You're going to hear that a lot over the next 11 sessions. One little defect in biochemistry, here comes the pathology. Here comes the pathology. All because what is pathology? Something going wrong with the biochemistry. And in sickle cell anemia, the best example, when the DNA polymerase did not fix and pause and check and correct itself like it should. So the DNA polymerase is high fidelity, but not perfect fidelity. All right, now let me ask you this. Why is it that RNA polymerase is low fidelity? Why is it permitted to be low fidelity? Why? Why do you think RNA polymerase, no big deal. Okay, so it makes a mistake. It's not corrected. What's the big deal? No, Luis, good try, Luis. It could be corrected. No, it's not corrected. Good try, though. At least you're trying. I appreciate that. Not permanent, that's true. And the reason is, so the idea is the RNA being made has a very short half-life. And RNA is not inherited, but it has a very short half-life, and it's turned over. New RNA is going to be made by RNA polymerase, and hopefully it has the right sequence. But the important feature is this is happening in one cell of one organ of the body. in that one cell, whether it was a hepatocyte or a muscle cell. Okay, you make a defective protein, big deal. It's not inherited. That one cell, the RNA will be turned over, then you make the right protein. So that is why RNA polymerase is permitted to be low fidelity, because it's happening in one cell of the organ, and it's not inherited. That's why. It just makes sense, right? It makes sense. Why one's high fidelity, the other is low fidelity. All right, now before we move on, question for you. You just had microbiology with Dr. Kim. All right, here comes the question for you. You should all get this one correct. Here comes the question. When you talked about HIV, you talked about a very important enzyme called reverse transcriptase. Here comes the question. Does reverse transcriptase, does it require a primer? You got a 50% probability of guessing the right answer. It's either yes or it is no. What is the answer? Does it require a primer? Let me see your answers here. Okay, I see mostly no's and some yeses, and the answer is yes, it requires a primer. What's being made? What's being made by reverse transcriptase? DNA. So reverse transcriptase is a DNA polymerase. It reads RNA, but what's being made is DNA. All DNA polymerases require primers, including reverse transcriptase, because it's a DNA polymerase. Now, again, notice how a little bit of information on what a DNA polymerase does, you could have got this answer in microbiology. But the idea here is this will be important several sessions from now once we talk about the PCR reaction. When we talk about polymerase chain reaction, we take a little bit of DNA and you make more DNA in a test tube. The DNA polymerase requires primers. So whether you need primers in a test tube or primers inside the cell, DNA polymerases, all of them require primers. Okay, there it is. Okay, now what's coming up next is the issue of what you need to know and don't not know about replication, the process of replication. Now, in your book on page 22 is a tabular way of discussing replication in prokaryotes and in eukaryotes. In your book, I think page 23, there's a diagram that says the same thing. I'm going to talk about it only in this table. Now, in this table, there's a lot of low yield information. And this column here is what happens in prokaryotes that you just talked about with Dr. Kim, and this right side is what happens in me and you. Now, of all the stuff in this table, let me show you right at the beginning what's low yield. On this figure, there's some high yield, extremely high yield stuff. But what's low yield? Here come, get ready with that. Let me show you what's low yield. Right there. The bulk of this material is low yield. I'm not going to cover it. The idea of how helicase unwinds a DNA, single-stranded DNA binding protein binds and stabilizes, and how you get leading strands. Yeah, you could look it up. It's in your book. You can look in first aid. It's low yield. I have not heard about that being on board exams in recent years. Low yield. Take a chance that it won't be on the, I cannot guarantee it, that it won't be on the boards, but take a chance. Focus your attention on the high yield material. What's high yield? It's coming up. The low yield, don't worry about it. Now, if you have to know it, if you're a type A personality and you got to know everything, help yourself. It's in the book, it's in other books, first aid, go read about it. Go ahead, I'm not stopping you, but you're wasting time. Focus your attention on the high yield material. 90% of the board exams, whether it's the USMLEs, whether it's COMLEX, whether it's Canadian boards or whatever, 90% of the exams is high yield integrated stuff. Integration of pharm, path, and biochemistry, physiology, integrated stuff. Low yield stuff, how does the leading strand, that's straightforward biochemistry. What does that got to do with pharmacology with it? It's low yield. So don't worry about low yield material. Take a chance. Take a chance, okay? Focus in on the high yield stuff. But if you want to know, help yourself. Nobody's stopping you if you're a type A personality. Take it. but you're wasting valuable time. All right. If they're in this low yield material, if there's anything that might be medium yield, that might be on board exams, there is an enzyme called 5' exonuclease in both prokaryotes and in eukaryotes. And what does the enzyme do? It removes the RNA primer. So the RNA primer is not found in chromosomes, so you have to remove it by an enzyme called 5' exonuclease. And even that, in my opinion, is not that high yield. So medium yield at best. Other enzymes, don't worry about them. If you want to know Pol-3, go ahead, but you're wasting time. That's my job, right? Is to tell you what you need to know and what you don't need to know. Now, at the bottom of the figure is the high yield material. There are two enzymes that you absolutely must know. Extremely high yield integration with other stuff. And one of them is called topoisomerase 2 in both prokaryotes and eukaryotes. We'll talk about that enzyme in just a few minutes. And then there's another enzyme in eukaryotes called telomerase. Okay, now what does telomerase do? As simple as I can state, is that when DNA polymerase binds to your chromosomal DNA and starts replicating the DNA to make more DNA, the DNA polymerase, as simple as I can state, the DNA polymerase does not finish what it started. And so if, just picture what would happen if that's all there was, and the DNA polymerase does not finish what it started, and so suppose that's going on in the germline of some individual. What that would mean is, in the sperm or the egg of that individual, the chromosomes will be shorter than what you have, if that happens to me and you. And so then in that progeny's progeny, they would be shorter chromosomes than the progeny had. So you would imagine in successive generations, the chromosomes would get shorter and shorter and shorter if the DNA polymerase does not finish what it started. That's a problem. You can imagine after many generations, there'll be no more chromosomal DNA anymore. And there's no more thing called, what's it called again? Life. There'll be no more thing called life. If the chromosomes, there is no more chromosomal DNA. So the body says, we got to do something about this. And what that body has is a major enzyme called telomerase. Now, what telomerase does, it works on the end of the chromosome. The end of the chromosome is called the telomere region. So because your chromosomal DNA is linear, the end of the chromosome is called the telomere. And what the enzyme does, telomerase, it just finishes the job that DNA polymerase started. So DNA polymerase starts the replication, it stops, and then telomerase says, okay, DNA polymerase, go to bed, I'll take over, I'll finish the job. And sure enough, that's what telomerase does. It prevents the chromosomes from shortening with increasing numbers of replications. Now, don't go worrying about the biochemistry, the mechanism, how this happens. People won Nobel Prizes for the mechanism. They're not going to ask you that, the mechanism, how this works. Don't worry about that. What's important to know, though, what's important to know about telomerase is, one of several things you need to know about telomerase is, this is a major example of the body that has reverse transcriptase activity. Even though the mechanism is not important, they might ask you, and I heard about this being on boards from students, they could ask you that telomerase, they might ask you which of the following enzymes has reverse transcriptase activity in the human body, and it is telomerase. So that's what you need to know. One of the rare examples of reverse transcriptase that's normal in the human body. All right, now another item here. It says in prokaryotes, it says a telomerase is not required. Let me see your answers here. Why is telomerase in prokaryotes, from Dr. Kim's review, why is it not required? Why? Why is it not required? Everybody should be getting this one. Since you just had the review in micro. That's right, it's circular DNA. Microbes, remember, have one chromosome. It's a circular chromosome. There's no end of the chromosome. So that's why in prokaryotes, there's no telomerase activity that's required. It just makes sense. Now, here comes another reason why you need to know telomerase. The integration, not with microbiology, but with pathology. In pathology, let me ask you this. In pathology, what's the name of the class of diseases in which in the cells they have increased telomerase activity compared to normal. It's not the cause of the disease. It's associated with the disease. They have increased telomerase activity compared to normal. What kind of diseases have that? That's right. Well done, everybody. That's right. Without looking at other people's responses, you'll say cancer. Cancer cells notoriously have elevated telomerase compared to normal. It's not the cause of the disease. It's associated with the disease because cancer cells are rapidly growing cells. And because the cancer progeny cells want to have the same chromosome length compared to the precursor cells, then they have elevated telomerase activity compared to normal. Now, another way they can ask you about this increased telomerase, normal cells in the body, like embryonic stem cells that are rapidly growing in the fetus and so on, those cells also have increased telomerase activity compared to normal. So any cell in the body that's rapidly growing, that's normal or cancerous, would have increased telomerase activity compared to normal if it's rapidly growing cell. All right, here comes another question. In pathology, what's the name of the disease in which the cells of the body have decreased telomerase activity compared to normal? Decreased telomerase activity compared to normal. Decrease. Not the cause of the disease. It's not the cause of the disease. Atrophy, there. Omar, there we go. By the way, if I mispronounce your name, I apologize. That's why I wish I could pronounce people's names better. I'm not good at that. So I apologize if I mispronounce your name. I like simple names like Sam, Bill, Tom, this type of thing, Beth, but if I mispronounce your name, I apologize. But in any case, the answer is indeed it's progeria. Remember, progeria is premature aging. These kids may be 8 years of age, but they look like they're 70 years of age. Premature aging, where the ends of the chromosomes are abnormal, and causing, related to that is they have decreased telomerase activity compared to normal. I want to stress it's not the cause of the disease. It's associated with the disease. All right, now notice here at the bottom of the figure, why do you need to know telomerase? That's high yield. Master it tonight or tomorrow at the latest. Master it. Understand why it's high yield. The integration of biochemistry with microbiology, with reverse transcriptase, and the issue of pathology. All right, now this other enzyme, topoisomerase 2. Why is that enzyme so important? Because of the integration with pharmacology and microbiology. Now, in pharmacology and microbiology, there is a class of drugs that you must know, high yield, and they are the fluoroquinolones, the new generation of the quinolones, drugs like Cipro and Levo, and what they do is they target topoisomerase 2 of prokaryotes, also called DNA gyrase. Now, don't worry about how topoisomerase 2 works. I'm not going to ask you about the biochemistry. The idea is that when DNA is replicated in bacteria, the DNA over-tightens, and you need topoisomerase 2 to relax the DNA, to loosen the DNA. That's all you need to know without the gory details. And so this happens in microbes. And what the fluoroquinolones do, they inhibit topoisomerase 2, preventing relaxation of the DNA, which is now over-tightened, and the bacteria cannot replicate DNA when it's over-tightened. And therefore, by inhibiting topoisomerase 2, they can't replicate DNA. Eventually, they will be eradicated. That's how they work. That's the target of the pharmacology, to treat the microbiology, to treat the pathologies and infectious diseases. All right, now that is, I didn't ask you about the fluoroquinolones because I know it's in the margin of your book on page, I guess 22 there, or 23. That's one of the nice things about being a participant in writing the book, a co-author of the book. I know what's in the books. I didn't ask you because I know it's right there. But what's not in the book, here it comes, in pharmacology. There is another class of drugs out there. And these drugs are drugs like etoposide and teniposide. What do those drugs do, and what do they target? Obviously something to do with this page, right? If these drugs were inhibiting HMG core reductase and cholesterol synthesis, we'll talk about that next week. What do these drugs have something to do with this page? What are they targeting on this page? Yeah, they're leukemic drugs. These are anti-tumor drugs, anti-neoplastic drugs, but what do they target? What are they targeting? You got to know the biochemistry. Yes, in pharmacology and pathology, they might ask, what drug you use, but they might ask, what was the target of that particular drug? And here it comes. It's topoisomerase 2 of cancer cells there. So if you inhibit the topoisomerase 2, because again, eukaryotic cells, when they replicate DNA, the DNA over tightens, supercoils. And what topoisomerase 2 does, it just relaxes the DNA. And now if cancer cells are replicating DNA, they get a lot of over tightened DNA, they have to relax the DNA with their topoisomerase 2. And in the presence of etoposide, topoisomerase 2 doesn't work. Cancer cells eventually will be eradicated because they cannot multiply. Now, importantly, in pharmacology and so on, let me ask you this. Is the enzyme in topoisomerase 2, the one that's in microbes, the one that's a target of the fluoroquinolones, that's a much different enzyme that's in eukaryotes. That's why the fluoroquinolones have no effect on me and you, because the bacteria have a different topoisomerase 2. But on the right side, the topoisomerase 2 that's in cancer cells, is it the same or different enzyme that's in normal eukaryotic cells? Let me see your answer here. Is the topoisomerase 2 that's in cancer cells, is it normal? Is it the same or different? Okay, very good. And the answer is it's the same enzyme. The topoisomerase 2 that's in a cancer cell is the same enzyme that's in a normal cell. Okay, here it comes. You know I'm going to ask you the why question. You get ready for the why. Then why do you give the drug? Etoposide and teniposide to treat certain cancers. Why do you give the drug if the normal cells have the same enzyme that the cancer cell has? Let me see your answers. They replicate rate faster. No, not quite the answer. Rapidly growing cell. Think cell cycle. Always think cell cycle here. The idea is cancer cells are in G1 phase. They're always growing. So they have a lot of topoisomerase 2. But most of the cells in the human body that are normal are G0 phase cells. Topoisomerase 2 is present. It's sitting there in the nucleus, but doing nothing. Just sitting around, having a good time. But it's doing nothing because the cells are not replicating because they're in G0 phase. Here it comes. In pharmacology, what are the side effects when you give somebody etoposide or teniposide? What are the side effects that are seen in an individual taking an anti-neoplastic drug like etoposide? Everybody knows this. What's the side effects? That's right. Hair loss, bleeding problems, GI tract problems. How does the biochemistry explains the side effects in pharmacology? And you'll get, if you think, you'll get the answer. Because cells in G1 phase that are normal, epithelial cells, topoisomerase 2 will not work in the presence of etoposide. Therefore, you're going to have GI tract problems. Nucleated red cells cannot multiply, and as a result, in the presence of etoposide, therefore, you're going to have bleeding problems. Follicle cells, G1 phase cells, will not be able to grow in the presence of etoposide because topoisomerase 2 is inhibited. Therefore, hair loss. It makes sense if you think about it. You can memorize, but it makes sense. Why? If you know the biochemistry. There it is. That's right. So again, look how beautiful this is. A little bit of biochemistry explains the form, explains the pathology, if you know what goes on here. All right, now what's coming up next is, so again, at the bottom of the figure, focus your attention on that. Master it as sooner the better, because you know, when Dr. Barone is here 2 weeks from now, he's going to be talking about cancer cells and progeria. You know what's the telomerase issue. When Dr. Harris is here in pharmacology, he starts talking about the fluoroquinolones and etoposide anti-tumor drugs. You could yawn because you heard it. You know what he's talking about. He's just going to reinforce it from another point of view, from pharmacology, but it all makes sense. Integration, integration, integration is what's high yield. Two or more disciplines is high yield for board exam. You're not going to see a straightforward biochemistry question. You're not going to see a straightforward micro question. Usually they're going to be integrated questions. Every so often I'll go on the internet and I look at these student doctor forums to get a big laugh sometimes, because what happens is a lot of students are giving misinformation to other students. Oh, biochemistry, don't worry about it. Don't worry about biochemistry. You got to worry about the 3 Ps, physiology, pharm, and path, and all this kind of stuff. But it's how the questions are worded. One time at my institution, there was a very bright student, and just a couple years ago, and after she took the exam, the U.S. boards, I saw her in the hall, and I said, "Hey, how you doing?" She said, I did pretty good on the board exams. I feel really good about it." And I said, "How much biochemistry did you have?" And she says, "Oh, man, I didn't have very much biochemistry at all. I studied all your stuff. Man, in the first year of medical school, I studied all your stuff. I had no biochemistry." And I said, "Really? You had no biochemistry?" She said, "Nope, I wasted my time." I said, "You mean to tell me, out of 322 questions, I picked a topic? You mean out of 322 questions, you had no question that dealt with," I picked the topic, "cholesterol metabolism"? She said, "Oh yeah, that's right. I did have a question about statin drugs in the treatment of hypercholesterolemia." "Well, that's biochemistry and pharm." And I picked another topic. "Did you have a question about iron metabolism?" "Oh yeah, that's right. I did have a question about hemochromatosis." "That's biochemistry and path." It's how you view the question. The biochemistry is there. You may not see it, but it's there. Every discipline depends on biochemistry. It's there. So all these people give misinformation about, biochemistry, don't worry about it. They're hurting. I'm telling you right now, they got their work cut out for you, because it's pure memorization at that point. All you got to do is think, and you'll do well on board exams. All right, now what's coming up next? High yield topic. You think the bottom of the figure is high yield here on page 22? Wait to the next figure. And this has to do with DNA repair. Now, on this table, there's some very high yield items coming up here. Pay attention, because Dr. Barone will also talk about the pathologies here, and you've got to know them right now as well. All right, now what's happening here is that under UV radiation, if you go outside on a sunny day, like here in Kentucky, it was a beautiful day today, like 80 degrees, beautiful day. If I'm working outside, I'm exposed to UV radiation. And what happens there is UV radiation will form thymine dimers. Two thymines on the same strand form a dimer. That's our problem. But by the way, you don't have to go outside. Even in your room right now, wherever you're located, the computer is generating a little bit, not much compared to outside, but there's a little bit of UV radiation that goes on in the room that you're at. Not much, but a little bit. And so your cheeks and so on are being exposed to a little bit of UV radiation. And you must be able to fix that problem. DNA repair. There is a major enzyme called excision endonuclease, and associated with a major disease called xeroderma pigmentosum. Now, in terms of the biochemistry, what does the enzyme do? Look at the words. Excision means cutting. Endonuclease means internally. What the enzyme does in your cells, it takes out the thymine dimer, but it doesn't fix the problem. DNA polymerase, DNA ligase then goes in and fixes the problem. One more time. Excision endonuclease takes out the problem. DNA polymerase and DNA ligase then goes in and fixes the problem here. So this enzyme, excision endonuclease, now, by the way, the mechanism, how the enzyme works, is in the next page. Don't worry about the mechanism. They're not going to ask you about the straightforward biochemistry. What they're going to ask you about is the integration with pathology. And that is this well-known disease called xeroderma pigmentosum. I don't know why, but this disease in pathology seems to be on the boards every year. I hear about it year in and year out. They're either asking you about the name of the pathology or the name of the enzyme that gives rise to the pathology. Now, how might they? Now, you should always, I'm not sure what the other instructors mentioned to you, But you should, when you get a vignette on board exams, always pay attention to the first phrase, because in the first phrase they might give you a clue of what's going on. You should always pay attention to the age of an individual. You should always pay attention to if it's male or female, because it might have something to do with X-linked recessive or X-linked dominant, something like that. You should always pay attention to the ethnic group or a geographical group. If it's like if it's some disease in the Mediterranean countries, you think, oh, they might ask me about G6PD deficiency, something like that. But here, always pay attention to the first phrase, because they might be giving you a clue. Here it is. Suppose they say to you on board exams, a light-skinned child. Automatically, that should be ringing bells. Light-skinned child. Then they continue on with the sentence. A light-skinned child with extreme freckling, lesions on the face, skin ulcerations. You automatically think, oh, they're probably asking me what's the name of the pathology, or they might be asking me what enzyme might be defective. So they can either be asking you about the pathology or the biochemistry, what's causing the pathology in this particular individual. So again, light skin, so what that means, and so geographically, they talk about somebody from Scandinavia and so on, always susceptible to UV radiation. It can end up with a lot of lesions where excision endonuclease might be a serious problem if it's defective in the Scandinavian countries. Major. This is how they do it on board exams. All right, now the next disease, high yield disease, is when we have now again, thymine dimer repair occurs in G1 phase. In G2 phase, we can have mismatched bases that can occur. And usually these mismatched bases occur in regions of the chromosomes which have microsatellites. Now later on, in chapter 7, we're going to talk about what microsatellites are. We'll talk about those also in genetics. Don't worry about them now. But these are regions of the DNA which have microsatellites, is where mismatched bases often occur. Fortunately for me and you, when we have mismatched bases in G2 phase, there are a pair of genes, and you must know these genes. One's called MSH2, the other is called MLH1. The little h refers to the human version of MSH2 and the human version of MLH1. Now, don't worry about what enzyme or protein is encoded by the gene. All you need to know for the boards is one of these genes encodes for a protein which finds the mismatched base. The other gene encodes for a protein which takes out the mismatched base. And then DNA polymerase, DNA ligase then goes in and fixes the problem. Now, why is this high yield? Okay, it's a pair of genes. Why is that so important? And the reason is because of pathology. And when either of these two genes is defective, any gene can be mutated. A lot to say about mutations later on. But if either of the genes are mutated, it gives rise to a well-known disease in pathology called hereditary non-polyposis colorectal cancer. Let me see your answers here. In pathology, what is the other name of this disease called? What is the other name of this disease called? That's right. Well done, everybody. It's called Lynch syndrome. So on board exams, either name is valid. So that's why you should always be cautious, because on board exams they might ask you about a certain disease, and you might think, I never heard of that disease, because they might be using a different name. So diseases in pathology have different names. Enzymes can have different names. Hormones can have different names. For example, calcitriol and 1,25-dihydroxycholecalciferol. It's the same hormone, but different names for the same thing. So you always got to be ready for boards which name they might use. So here it's Lynch syndrome. The other name for it is hereditary non-polyposis colorectal cancer here. Again, notice that these cells are in G1 phase. They're growing. And so the idea is, why is it causing cancers and so on? And the reason is when you have a mismatched base, and if it's not corrected, what happens to the double-stranded DNA? It's not very stable. Because of the mismatched base, DNA is unstable, and DNA can fragment. the chromosome might break. So the way they might do this is, on boards, they might say to you, a biopsy of the colon was taken. And when they looked under the microscope and did some sort of karyotype, something like that, when they looked at the chromosomes, they found that some of the chromosomes looked like they were broken. They looked like they were fragmented. If it came from the colon, you say, wow, they're probably asking me about Lynch syndrome. Or they might be asking, what's the name of the disease? What's the name of the genes that might be defective? Or what's causing it? And you say, well, it's DNA instability, something like that. Different ways to ask a question about that vignette. So please, always try to anticipate, how could they ask me about a certain item? And it's usually integrated items. Now, at the bottom of the figure, again, Dr. Barone certainly is going to be talking about Lynch syndrome in his great lecture. So when he starts talking about it, you know what he's talking about. There's no mystery here. Now, at the bottom of the figure is much lower yield. This is called cytosine deamination, which occurs in G1 phase. Now, without looking, don't go looking. When this occurs by spontaneous means, heat or nitrates, if you eat a lot of hot dogs, there's a lot of nitrates in hot dogs. And the reason is because nitrates are meat preservatives. So hot dogs have meat preservatives called nitrates, which try to stabilize the degradation of the meat. But the problem is, if you eat too many hot dogs, you're eating too much nitrates, and nitrates can deaminate cytosines in your DNA. Now let me ask you this, without looking, don't go looking. What's the name, when cytosine is deaminated, what does it form? When cytosine is deaminated by nitrates, what does it form? That's right. See that? You're a thinker. That's right. Thank you. Thank you. It's uracil. And sure enough, you would anticipate there is an enzyme called uracil glycosylase that removes the uracil, because uracil should not be in DNA. Now, don't worry about the mechanism. There's an enzyme called AP endonuclease. Don't worry about that. And then DNA polymerase, DNA ligase then goes and fixes the problem here. All right, so again, you should know the pathologies, and you should know where in the cell cycle these types of repair mechanisms occur. Now, I got another question for you before we leave page 24. Get ready with your fingers. Is there any repair that occurs in S phase, in the cell cycle? Now think, don't go turning pages. Is there any repair occurs in S phase? Yes? Okay, and if you say yes or no, if you say yes, what is it? What is the repair that occurs in S phase? It's the proofreading enzyme, that's right, but more specifically called what? What's it specifically called? In DNA polymerase, it has exonuclease activity. Is it the 5' or is it the 3' exonuclease? Without looking, I'm trying to get you to recall. Is it the 5' or is it the 3'? And the answer is, it's the 3' exonuclease. So the 3' exonuclease is part of DNA polymerase, that what it does, it takes out the incorrect nucleotide in S phase. By the way, what does the 5' exonuclease do? Without looking, don't go looking. What did the 5' exonuclease do? Just to compare. 3' exonuclease, what was the 5'? That's right. Yeah, well done, everybody. It removes the RNA primer. That's right. In replication, it removes the RNA primer, starting with a 5' end. But notice over here, notice that the 3' exonuclease is the primary way to repair DNA in S phase, while the chromosomal DNA is being replicated. And when the DNA is out of S phase, when the cells are in G2 phase or when they're sitting in G1 phase, they can be damaged. UV radiation, chemicals in the environment can damage the DNA. And fortunately, we have repair mechanisms because the body is going all out to make sure the chromosomes that you pass on to your progeny minimize the mutations that are in them. So again, the concept, part of the bigger picture, you inherited chromosomes from your father and your mother, and you're going to pass them on to your progeny. And your body is trying everything to do to make sure the DNA sequences of all 46 chromosomes that you're going to pass on, 23 chromosomes onto your progeny, are going to be the right DNA sequence without mutations. So this is the primary way to remove mutations, And these are secondary mechanisms, the thymine dimer repair, the mismatch base repair, and now we come to the ultimate repair mechanism. The ultimate repair mechanism, high yield. And that is this all-important topic called tumor suppressor genes that Dr. Barone certainly is going to be talking about when he's here. Now, what this is about is that cells that sit in G1 phase. Remember, epithelial cells are sitting in G1 phase. They can be exposed to chemicals in the environment like benzene, UV radiation from the sun. You go through a metal detector at the airport, you get radiation. You go get a chest x-ray at the doctor or something like that. You get your teeth examined by the dentist and you're x-raying. You're damaging some cells. And so the idea is cells as they sit in G1 phase, waiting for the signal, waiting for the hormone to tell the cell to start growing. As they sit there in G1 phase, they can be damaged. And so what happens is, the example I'm going to talk about here is P53. This is a gene called P53 that encodes for a protein called P53. P stands for protein, 53 is the molecular weight. And what P53 is, it's good for me and you because it sits at this green line, the boundary between G1 and S phase, and it sits there as it's like a sentry. It's a sentry. It sits there watching cells go from G1 into S phase like a sentry. And when a cell is damaged, DNA, P53 will prevent that cell from going into S phase. Here comes the question. Like a traffic cop. That's right, Daniel. That's like a traffic cop. You can go. Oh, you cannot go because you have damaged DNA. Here comes the question. How does it prevent those cells from going into S phase with damaged DNA? How? Let me see your answers here. How does it prevent the cells from going into S phase if it has damaged DNA? Slows the cycle? No. Good try, Vincent. At least you're trying. And the answer is it kills the cells. That's pretty rough treatment, right? That's all for you. You're done. Get out of here. I'm going to kill you. And sure enough, there it is. It does so by the process of apoptosis. So again, the tumor suppressor genes, this example is P53. It prevents the cells with damaged DNA from going to S phase, and it does so by killing the cell by the mechanism called apoptosis. And why P53 is good for me and you? Because if it's not killed, the cells go into S phase, and as a result, they end up with lots of damaged DNA progeny cells, and that causes tumors. So why is P53 good for me and you? Because it suppresses tumor formation. So all of these tumor suppressor genes, like P53, ATM, BRCA1, BRCA2, the retinoblastoma gene, all these genes, they work the same, but they're in different locations, whether in the retina or whatever, epithelial cells, they're in different locations, but they work biochemically the same. They act as traffic cops, as Daniel mentioned, or sentries, and they watch cells, and when they have damaged DNA, they kill the cell by the mechanism of apoptosis. All tumor suppressor genes are good for me and you. But like anything else, any gene can be mutated. And if the gene, in this example for P53, is mutated, now think what's going to happen. Just think, if the gene for P53 is damaged, it has a mutation. Then cells with damaged DNA in G1 phase because of exposure to the environment, now they're no longer being killed off, and they go into S phase. They start replicating when they should not, giving rise to Lee-Fraumeni syndrome, and they're characterized by many solid tumors. There it is. You know you're going to see this from Dr. Barone, but you know now the why. Many solid tumors. Because cells with damaged DNA go into S phase, they start replicating, multiplying, many solid tumors. Lee-Fraumeni syndrome. Now again, when Dr. Barone's here, he'll talk about ATM and BRCA1, the retinoblastoma and all that kind of thing. But the biochemistry is all the same. The locations, the pathology as a result is different. but it's the same principle. Make things simple for yourself. Understand concepts. Understand. Don't make it difficult. Just understand the bigger picture here. Okay, everybody, perfect timing. It's 6 minutes to the hour, so I'm going to take our last 10-minute break. We're done with chapter 2. There it is. Everything you need to know about replication was just discussed. Everything. So we're done. No reason to read about it, unless you want to. Everything was just discussed. So as long as you were paying attention, you know what's high yield. Focus your attention on that. You don't have to worry about replication because you mastered the high yield material. Okay, I'll be back in 10. Welcome back, everybody. And so we're done with chapter 2, and now we're going to get into chapter 3. So chapter 1, what have we done tonight? So tonight we talked about what is a chromosome, what's DNA, what's RNA, what are the precursors to make them. Chapter 2 dealt with DNA replication, meaning DNA synthesis. So again, as long as you were paying attention, as long as you're focusing, as long as I did my job properly, there is no reason to go reading about it, wasting time reading other books. As long as you were understanding what I said, it's all the high yield stuff was discussed. Now, if you want to know more, that's great. But if you don't, but as long as you were understanding, all the high yield material was covered. Now, again, I know some of you still fear biochemistry, but don't fear it. Make biochemistry your friend. Biochemistry wants to help you. Biochemistry wants to walk with you. Biochemistry says, hey, please don't kick me away. Don't kick me away. Help. I want to help you. Biochemistry wants to help. Give it a chance. Understand the concepts, and it will help you knowing what's going on with the other disciplines. Don't fear biochemistry. Be positive. Stay positive. Always say to yourself, what's in this that's going to be useful? What do I need to know? Replication. We just talked about it. DNA repair, the diseases, the pharmacology with the quinolones. It's already been reviewed. So the idea is, what's in it for me? Why do I need to know it? Make biochemistry your friend. Okay? And I'm telling you right now, you're going to be so much better off. Make it your friend. Okay? Don't kick it away. It wants to help you. All right, now the next chapter to talk about is transcription. Now you know right at the beginning, if I ask you what does transcription mean, you all say RNA synthesis. So that's what's going to be discussed here is RNA synthesis. Now after we do the basic biochemistry, be ready for a lot of pathology. There's going to be pharmacology as well. Major stuff coming up in chapter 3. A lot of things go wrong in eukaryotes regarding RNA synthesis, but we've got a long way to go before we get to the high yield items. Now, let me ask you this: if I were to ask you, what does DNA? Without looking, don't go looking now. Don't go turn. Hey, I hear somebody turning the pages. Don't go turning up. Okay, nobody turning pages. All right. Now, if I were to ask you, what does DNA polymerase have to see on the DNA to say, I got to sit down on this DNA to begin the process of replication? What's your answer? That's right. Everybody should say a primer. That's right. All right, well done. But now, but RNA polymerase does not require a primer. So what does the RNA polymerase have to see? And the answer to this is, what an RNA polymerase sees, never the gene. What RNA polymerase finds and binds to a promoter sequence, never the gene first. Now, the promoter is a DNA sequence which precedes the gene. So I want to stress, RNA polymerase never binds to the gene first. It binds to a sequence of DNA that precedes the gene called the promoter sequence. And later on, I'll show you the integration with pathology. You must understand the promoter. It is a DNA sequence which precedes a gene to which RNA polymerase binds and begins the process of RNA synthesis. Now, regarding the gene is double-stranded DNA, but only one of the two strands is used as a template by the RNA polymerase. The other strand basically is useless, but only one of the two strands is used as a template. Now, because one strand is used by RNA polymerase and the other strand is not used, both strands of the RNA are given names. Let me ask you this. What is the name of the RNA strand what is the name of the DNA strand of the gene that's used as a template by RNA polymerase called? What's the name of the strand that's used as a template? Man, why not call it the template strand? Wow. I wonder where that name came from. The template strand is used as a template by RNA polymerase, and it's going to be complementary and anti-parallel to the RNA being made. Now, what's the name of the other strand that's not used as a template called? What's the name of the other strand that's not used as a template? That's right, it's called the coding strand. The coding strand is not used by RNA polymerase, but this is key. Underline this, please. It is identical. I'll show you how important this is in a few minutes. The coding strand is identical in sequence to the messenger RNA being made, with the exception of T's and U's. The coding strand has T's, the RNA has U's, but other than that, it is the same sequence. It's identical in sequence to the RNA being made. And I will show you how important this is in just a few minutes. Now, in the yellow box coming up, nothing new. We talked about it earlier. Is DNA, the gene is read, DNA gene is read 3' to 5' by RNA polymerase, and RNA is made 5' to 3'. Nothing new. We talked about that earlier on page 19. Nothing new about this yellow box. Now, suppose on board exams they give you this diagram at the bottom of page 33. They say to you, this is gene 1, this is spacer DNA, gene 2, spacer DNA, gene 3, more spacer DNA. Now, let's focus our attention on gene 1. Now, suppose they say to you, they give you the information as shown by the black arrow, that they say to you the direction of transcription is from your left to your right. So that's the direction of transcription. In gene 1, upper strand. Is that the coding strand or is that the template strand? They say to you direction of transcription is left to right. Is that the coding or is that the template strand? Okay, and the answer is it's the coding strand. And the lower strand is the template strand. Now, how do we know that? Because it says the upper strand's 5' to 3', the lower strand is 3' to 5'. And remember, DNA is red, the template strand is red, shown here in the yellow rectangle, 3' to 5'. So what's going to happen is RNA polymerase will bind to the promoter, shown here in the green, just upstream, a DNA sequence to which RNA polymerase binds. And then it's going to read the template strand 3', there it is. This is the 3' end of gene 1. Here's the 5' end of gene 1. It's going to read it left to right in this example, making RNA as a product because it says direction transcription is left to right. Same thing with gene 2. Now, suppose on board exams they talk about gene 3, and suppose they say the direction of transcription is from right to left. In gene 3, upper strand, is that the coding strand or is that the template strand? Let me see your answers. Upper strand with gene 3, coding or template? That's right. Well done, everybody. That is the template strand. And the lower strand is coding. Because on the upper strand, this is the 3' end. This is the 5' end of gene 3. So the RNA polymerase will read it, bind to the promoter just upstream, and read it 3' to 5', making RNA 5' to 3'. Notice in one chromosome, this is a part of a chromosomal DNA. In this part of the chromosome, the RNA polymerase can go in either direction. It can go in either direction. It all depends where the promoter is located. All right, now the kinds of RNA that can be made. There are 3 kinds of RNA that can be made. Messenger RNA, which would be the most of the topic in chapter 3. A lot to say about messenger RNA. We'll spend one figure talking about tRNAs. What do you need to know about tRNAs? That's coming up later on in chapter 3. And we'll talk about one figure dealing with ribosomal RNA. What do you need to know for the boards about ribosomal RNA? That's coming up a little bit later. All right, now another item here. Let me integrate this with cell biology and histology. If they were to ask you, where is most, look at the word, listen to the words, most. Where is most of the ribosomal RNA being made in the cell? Where? Be specific. Be specific. Where is most of the ribosomal RNA being made? Be specific. That's right, it's being made in the nucleolus. That's right, most of it, but not all of it. And where else is it being made, we'll talk about later on. but most of the ribosomal RNA is made in the nucleolus. Remember that single biggest spot in the nucleus way back on page 12? If they ask you what is it, you say that's the nucleolus. If they ask you what's happening there, you say, wow, that's where most of the ribosomal RNA is being made. All right, here comes the question. Next question. Where is most of the messenger RNA and the tRNA, transfer RNAs? Where are they mostly being made? Be specific. Be specific. No, not the ribosomes. No, no, no. Where are they being made? They're being used in ribosomes, but where are they being made? And it is in euchromatin. Remember the light staining spots of the nucleus? Remember when the DNA is exposed? That's when RNA polymerase can come in and make RNA. It can either make messenger RNA or ribosomal RNA. So if they ask you, which of the following molecules are being made in the light staining spots of the nucleus? While you know the light staining spots of the nucleus is euchromatin, if they ask you what's important about it, you say that's where you have active gene expression. If they ask you what molecules are being made, you say, well, that's where most, not all, most of the messenger RNA and tRNAs are being made. Exceptions to this we'll talk about later on. Don't worry about it now. But again, this is how they integrate the biochemistry with the histology and the cell biology. Where are things being made in the cell? Okay, now that somebody mentioned ribosomes, but that's what they're being used to make ribosomes. The ribosomal RNA is being used to make ribosomes, but where are they being made is in euchromatin. All right, now what's coming up next is I'm going to take gene 1 and now give it some more information, more detail with a particular gene, and actually show you nucleotides. That's what's shown on the next figure here. Suppose we have a particular gene in the blue. So in the upper part, this is the coding strand, 5' to 3' because it says so. And the other strand is a template strand, 3' to 5'. And notice how these two strands of the gene are complementary, A with a T, T with a A, G with a C, and so on. Complementary and anti-parallel. 5' to 3' upper, 3' to 5' lower. Nothing new. It should all make sense. You know that is double-stranded DNA. So what we're going to be talking about on this page 36 is flow of genetic information. How we go from a gene to RNA to protein. Flow of genetic information. Major concept. Not the detail. Don't worry about the detail. It's the concept to focus your attention on. All right, so what's going to happen is just upstream of the gene, there'll be a promoter, not shown here. Promoter serves as a binding site for RNA polymerase. RNA polymerase will bind to the promoter and read the template strand 3' to 5'. Nothing new. So the 3' deoxy TMP is made in the 5' AMP in the RNA. Deoxy AMP in the template strand of the gene is made in the UMP in the RNA. Here's a deoxy CMP in the gene, made in the GMP in the RNA. Notice the direction of transcription, always 5' to 3'. Again, nothing new. You should be yawning at me now. Oh yeah, he talks about that already. Nothing new. Okay, so RNA polymerase binds to the promoter, reads the template strand, making RNA in direction 5' to 3'. Okay, then what's going to happen is in chapter 4, later on tomorrow, we'll talk about how the messenger RNA will get into the ribosome. The ribosome will now start reading the RNA, and 3 nucleotides make up a codon, and a codon specifies an amino acid. In chapter 4, the beginning of chapter 4, I'll show you the genetic code, how you use the genetic code. 3 nucleotides make up a codon which specifies a particular amino acid. As you will see in the genetic code, the beginning of the next chapter, AUG specifies methionine, GGG in the genetic code specifies glycine, CUC specifies leucine, and so on. That's the direction of translation. Translation means protein synthesis. Proteins are always made starting with amino terminus, finishing with a carboxyl terminus. Now what you see here are 4 amino acids. But a protein might be made that may have 500 amino acids or 1,000 amino acids. It doesn't matter how big the protein is. You always start with amino terminus. The direction of translation, always the amino terminus. And the first amino acid is always called position 1. The second amino acid is position 2. The third one is 3, and so on, all the way to the end. So this is important. So if on board exams, if they say to you, as an example, if they say to you, by the way, one more item here. Notice how the RNA is always 5' to 3', is being read by the ribosome, always on the 5' to 3' direction. So the direction of translation by the ribosome, it binds to the RNA and starts reading it 5' to 3'. So the 5' end of the RNA always encodes position 1 of the protein. Now let me illustrate how important this is, that you need to know. The RNA is being made 5' to 3', and the ribosomes jump on and it reads the messenger RNA 5' to 3', making the protein starting with the amino terminus and finishing with the carboxyl terminus. Now let me ask you this, as an example, illustrate this principle. In sickle cell anemia, the mutation is at codon 6. There's an A replaced by a T. And as a result, the amino acid glutamate is converted to valine because of one nucleotide change in beta globin at position 6. There's a glutamate converted to valine because of a one nucleotide change. And let me ask you this. Is that near the amino terminus of beta globin or near the C terminus of beta globin? Where is the mutation? If they say to you single digit amino acid, that's where mutation is. Is that near the N terminus or is that near the C terminus? It's near the amino terminus. Position 6 is near the amino terminus. Beta globin may have several hundred amino acids here, several hundred. If they say to you triple digits, like there's a mutation at position 282, you say, well, that's near the C terminus. So any single digits of a protein, it's near the N terminus. Now, those of you who said C terminus are thinking, well, if a protein's only 5 amino acids, there's no protein that's 5 amino acids. Most proteins are going to be hundreds, if not thousands, of amino acids. The chloride channel protein is like something like 520 amino acids, something like that. But the idea is, if it's single digits, you say, well, that's near the N terminus of the protein, near the 5' end of the messenger RNA. If they say to triple digits, position 282, you say, well, that's near the C terminus. Okay, you understand that. So please understand the numbers, because you might see that on board. You're going to see this later on in biochemistry and molecular biology. We'll talk about hemochromatosis. In hemochromatosis, the mutation is at position 282 of a particular protein causing hemochromatosis, and that's near the C terminus. Whereas in beta globin sickle cell anemia, that's near the N terminus, where the mutation happens to be. All right, now one more item here, one more item. So on board exams, what they might do after we do chapter 4, they might give you a sequence of double-stranded DNA, a gene, and they give you the genetic code. And they say to you, figure out the amino acid sequence. We'll practice this in chapter 4. Right now we're at the beginning of chapter 3. But the idea is, then you would imagine how much time this is going to waste. You figure out which one is a template strand, you draw out the messenger RNA, and then from the messenger RNA, you look at the genetic code to figure out the amino acid sequence. That takes a lot of time. But notice there's an easier way. Because if you're given a double-stranded gene, you look at the coding strand first and only. Look at the coding strand, even though it's useless by RNA polymerase. But notice the coding strand. I had you underline this on the previous page. Notice it's identical to the messenger RNA in sequence, with the exception of T's and U's. ATG in DNA means AUG in RNA. GGG in DNA means GGG in RNA. a CTC in DNA means CUC in RNA. Notice how the coding strand is identical to the messenger RNA. So if you're given the coding strand, you can locate the coding strand, you can deduce the amino acid sequence. And in fact, that's what's reported in databases. What I mean by this is, on board exams, and let me stress, please get this through your head. On board exams, if they give you a sequence of DNA, one strand, you must assume it's the coding strand, unless they say it's a template strand. But if they don't say anything about it, you must assume it's the coding strand. Because if you know the coding strand, you can deduce the amino acid sequence. What I mean by this is, when we're done today, I can go to my computer, my laptop that I'm talking to you right now. If I were to say, do a Google search and say, tell me the sequence of the human insulin gene, immediately by Google search on the monitor will come the insulin gene, and it will be one strand, and you must assume it's the coding strand for the insulin gene, because once you know the coding strand, easy to figure out the amino acid sequence. Again, please understand, it is always a coding strand unless they say in black and white it's a template strand. All right, now, eyes up, eyes up, eyes up. Let me see your eyes. Whew, you got beautiful, beautiful eyes. Okay, let me see your eyes. Don't go looking at the next page. Don't go look. Don't go looking. Promise me don't look at the next page. Because I'm going to show you the next figure coming up is a sample question coming from the next page. And on the next page is the answer. I don't want you to look at the answer, please. So try it on your own. No timer here, no selection pause. Just give you 30 seconds, look it over, come up with an answer on your own, and then we'll deduce the answer. Okay, but please, just try. Don't look at the answer. Just look at the monitor. Just simulate a board exam. Just look at the monitor here. Don't look at the next page. Here comes the sample question, okay? All right, don't go looking. Don't go. I know some of you say, oh, I want to look. Don't look, okay? Here comes the question. 30 seconds. Just look it over, and then we'll give you the answer. Oh, well, Beth put the selection pod up. Go ahead and, without looking. Now, if you look, don't put the answer. Okay. Okay. 10 more seconds. Don't go looking. Okay, and stop the poll and broadcast the results. And it looks like, let me broadcast it, there we go. Thank you, Beth. And it looks like slightly less than half the students put option D. A lot of students put option A and option E. Now you see why I'm doing the question. Okay, more than half the students got it wrong. And the answer to this question is option D. Option E. The majority of students got it wrong. The answer is option E here. Okay, let me move this out of the way. All right, now, this is, again, it's a matter of thinking. Now, those of you, a lot of you who got it wrong were trying to memorize. Just think. Okay, now, in this vignette is, all you got to do is, and they give you a vignette like this on boards, all you got to ask is, do they want DNA as an answer, or do they want RNA as an answer? And right at the beginning, they're telling you they want RNA as the answer, right? So if they want RNA as an answer, look here. It says cross out any option that has a T, because RNA doesn't have T in there, right? So, no, watch my PowerPoint. Watch. Get ready, everybody, eyes up, look at the monitor, look at your monitor, watch. We can now cross out any option that has a T. We can cross out option A, B, and C. Voila! Quickly wipe out option A, B, and C. Why? There are DNA answers. It wants RNA as an answer. So we can quickly eliminate option A, B, and C because those are DNA. Look how simple that was. So now we go from 5 possible options to 2 possible options. Then you can just guess. You got a 50% probability of guessing the right answer. All right, now what they're saying is, now again, this is how you, this next part here is why you must think and not memorize. So they're telling you there's a DNA template sequence, TAGC. Now remember, I told you this earlier. If no orientation is given, assume 5' to 3'. So they're saying to you, they're telling you it's a template sequence. They're giving you that information. It's a template sequence. All right, now let me ask you this. On the left side of this template sequence, the TAGC, on the left side, is that the 5'? Let me see your answers here. Is it the 5' end or the 3' end? Is on the template sequence, is that the 5' end or is that the 3' end? Okay, now I'm not looking. What does it say in the yellow box? I'm not looking. You tell me. Let me hear the answer. What does it say in the yellow box? Let me hear your answers. Don't go typing. Oh, yeah, that's right. It says in the yellow box, if no orientation is given, you assume 5' to 3'. There it is. Let me put it down. 5' to 3'. There it is, okay? I told you that earlier in the first hour. I said, be ready for this. A lot of you are going to make a mistake because you forgot I told you. No orientation is given, you assume 5' to 3'. Okay, so what does that mean? So it says a template sequence. What does that mean? That means an RNA polymerase, when it transcribes, makes RNA from that DNA template sequence, is RNA polymerase going to go from left to right or right to left? Which direction? Left to right or right to left? Which direction is it going to go? Left to right or right to left? No. It's going to read right to left. It's going to read the 3' C. Okay, so it reads the 3' C and makes it into what? It's going to make it into 5'. Here comes, watch, 5' G, C, U, A to the 3' end, because it's going from your right to your left. That's why you had to be here earlier and paying attention, because how the RNA polymerase works. So it makes this RNA as a product. There. Okay, so now why is the answer not D? Let me see your answer. Why? Let me see. Why is D incorrect? Let me see your answer. Let me see. Why is D not right? Because what does it say in the yellow box? Let me hear your answer. What does it say in the yellow box with option D? If no orientation is given, you must assume 5' to 3'. Notice that sequence there, 5' AUCG is not the same as 3' AUCG. So in other words, we take that sequence right here, shown by the green arrow, and flip it 180 degrees, and we now end up with option E, 5' G, 5' G, C, U, A to the 3' end. 5' G, C, U, A to the 3' end. It's not hard. Now, those of you who think, oh man, it's not hard. It's a matter of thinking. You got a beautiful mind. Use it. Use it. Dust it off. Use your mind. It's not hard. It's a matter of thinking. You can't memorize this. It's a matter of thinking. It's not hard. And if you get a question like this correct on boards, you got such an advantage, because so many people can get it wrong. You saw almost 3/4 of students got it wrong. This is after we reviewed the material. But if you think about it, oh yeah, it's not hard. It's a matter of thinking. Nothing tricky. It's not tricky. It's a matter of thinking. And now I'm going to try to get you to think and not memorize here. All right, now at the end of the chapter, there's another sample question. In fact, at the bottom of this figure, another sample question. Try it out on your own. Try it. Just do it a few times. Oh yeah, now I get it. Now I get it. Not hard. Just try it out. Okay. We got to move on, but there's one at the end of the chapter. Just try it out. If you have a question, just email me out at the very end. But try it out. We got to move on here. All right. Now, what's coming up next is what happens in prokaryotic transcription. Then after that, what happens in eukaryotes. Now, in eukaryotes, how you and I make RNA in our cells, very complicated. So because it's very complicated, be ready for a lot of pathology. Because when you make RNA, it's very difficult, and you're going to see a lot of pathology. In prokaryotes, it's much easier to understand. So what we're going to be talking about here is what happens in microbes. Now, Dr. Kim Muscatella may have talked about some of this stuff, but this is the biochemical point of view. So we're going to integrate the biochemistry with the micro, and then afterwards get ready for this thing called pharmacology. All right, now there's a term, definition that we need to address first, and that term is called UTR, which stands for untranslated region. What does that mean? As simple as I can state, the messenger RNA that's going to be made in both prokaryotes and in eukaryotes, not all of the messenger RNA will encode for protein. Every messenger RNA made in E. coli, as well as in your cells and mine, every messenger RNA has 5' UTR at the beginning of the RNA, will not encode for protein. Every messenger RNA will have 3' UTR. So 5' and 3' UTR, every messenger RNA in prokaryotes, eukaryotes have untranslated regions. Again, I want to stress that not all of the messenger RNA encodes for protein. And that which does not encode for protein is called UTR. Please understand this. And why is that so important? Because that's what Fragile X is about. Fragile X syndrome is a mutation in the 5' UTR. So please understand what UTR is. It's a region of messenger RNA on both the 5' end and on the 3' end that does not encode for protein. All right, now what I'm going to do over here is suppose in the orange, this represents some gene region in E. coli or some other microbe. And now what I'm going to do here is, we're going to make this gene into RNA in E. coli. Now what I'm going to do here is, the way I'm going to approach this is, I'm going to give you 6 numbers. After I discuss these 6 numbers in prokaryotes, I use the same 6 numbers to talk about what happens in me and you, in eukaryotes. Item number 1, in bacteria, there are things called operons. Operon, like the lac operon, histidine operon, what that means is polygene region, polycistronic messengers. Now, Dr. Kim may have talked about that earlier in microbes, but microbes have polygene regions here. Now, do you and I have operons? Stay tuned. That's going to be coming up 2 figures from now. But bacteria have lots of operons, lac operon, histidine operon, polygene regions, multiple genes in one region. Item number 2 of 6. Just upstream of the operon region in bacteria is the promoter region, shown here in the green. Don't worry about these negative numbers right now, but that's not that important. But the negative numbers are referring to nucleotides in the promoter region. So the negative numbers are referring to nucleotides. Increasing negative numbers are nucleotides of the promoter region. Increasing plus numbers are nucleotides of the gene region. So the -1, -2, -3, -4, -5. Increasing negative numbers are referring to nucleotides of the promoter region. Increasing plus nucleotides are referring to nucleotides of the gene region. So the +1 nucleotide, +1, +2, +3, +4, +5, +6, 7, 8, and so on, that's of the gene region. So the RNA polymerase now will bind to the promoter. It's going to read the template strand, in this example, the lower strand, because it's 3' to 5', and the +1 nucleotide of the template strand is made in the +1 nucleotide of the RNA. The +2 nucleotide, the +3 nucleotide, the +4, 5, 6, all the way. So the RNA is now getting bigger and bigger and bigger and bigger. Item number 3, in bacteria, there is no such thing as exons and introns. And because there isn't anything, we're not going to talk about them right now. We'll talk about them once we get to eukaryotes. But in bacteria, there's no such thing as introns or exons. Item number 4 is how does the RNA polymerase know to stop making RNA. So RNA polymerase is going to bind to the promoter, read the template strand 3' to 5', making RNA, and RNA is getting bigger and bigger and bigger. So the item number 4, how does the RNA polymerase know to terminate transcription? And the answer is, if I can use a little cord here, here's a cord. Suppose this white cord represents RNA being made, and suppose this, my fist, represents the enzyme RNA polymerase. So now, RNA polymerase will bind to the template strand and make RNA 5', here's the 5' end, making RNA 5' to 3' direction. So RNA is getting bigger and bigger and bigger until somewhere on the 3' end, there's going to be a stretch of C's and G's. A stretch of C's and a stretch of G's. And you know G base pairs with C. So on the 3' end, there's going to be base pairing of the C's and G's. This is sometimes called the stem loop sequence. A GC-rich C's and G's stem loop sequence. Now, why is that important? Because once that 3' end of the stem loop sequence is formed, it base pairs with itself, and that's like a 3-dimensional loop. And remember, RNA polymerase is a big protein, and what happens is when the stem loop sequence is formed, it gooses the RNA polymerase and tells the RNA polymerase, get off the DNA. So it gooses the RNA polymerase, and RNA polymerase comes off, just like that. One more time. So RNA polymerase binds to the promoter, starts making RNA 5' to 3' until somewhere there's a stretch of C's and G's on the 3' end. It forms the stem loop sequence and then gooses the RNA polymerase, and the RNA polymerase falls off, just like that. That's all you need to know. And so now here is the product. Notice it's here's the 5' to 3' end of the RNA. And here's the 5' UTR that will not encode for protein. Here is the reading frame in the orange. On the 3' end, there is that stem loop sequence, which for your purpose terminates transcription in prokaryotes. Now what's coming up next, I'm going to take this figure right here, the RNA, the messenger RNA that got made. I'm going to show it on the next figure and talk about items 5 and 6, the last 2 items. Let's go to the next figure. And again, remember, we're still talking about prokaryotes. Okay, now in the upper part of the figure, suppose this is an operon region. Here is multiple genes. Here's gene 1, spacer DNA, gene 2, spacer DNA, gene 3, and so on. And here's the promoter for all 3 of these genes. This could be the lac operon, for example. Remember, this is part of a circular chromosome in E. coli or some other microbe, a region of a big chromosome in a microbe. So again, remember at the top part of the figure, that's DNA, a region of the chromosome in some microbe. So RNA polymerase is going to bind to the promoter, and then RNA polymerase will now read the first nucleotide, the +1 nucleotide in the +1 nucleotide of the RNA, +2, +3. So RNA is now being made, but the RNA polymerase is getting bigger and bigger and bigger, and then until somewhere the stem loop sequence is encountered. And that now is RNA. Multiple genes in one region. Notice the RNA that got made. 5' UTR, here's gene 1 RNA, here's more spacer RNA, gene 2 RNA, more spacer RNA, junk DNA, or junk RNA I should say, more gene 3 RNA, and the 3' UTR. All right, so that, now item number 5. Again, this is, remember in item number 5, we get 6 items to talk about. Item number 5, there is no transcriptional processing in bacteria. Look at the words. No processing of the transcript. There's nothing like a poly A tail, nothing like splicing, none of that stuff happens in bacteria. It will in eukaryotes, but in bacteria, nothing. No transcriptional processing. So once we get to item number 5, in eukaryotes, see these 2 letters here, NO? You're going to save ink. You won't have to use your pen to write NO because item number 5 has transcriptional processing in eukaryotes, but not in prokaryotes. Now, item number 6, the last item. So remember from Dr. Muscatello's lectures and so on is in microbiology. Remember, bacteria have no organelles. There's no such thing as a mitochondria or nucleus in bacteria. So that single chromosome, that circular chromosome in bacteria is right there in the cytoplasm. So the RNA polymerase then transcribes the genes into the RNA and right there in the cytoplasm. And in the cytoplasm are the ribosomes. And the ribosomes now will jump on to that messenger RNA to translate the genes into protein. So if you were asked then, how do the ribosomes know to jump on to the bacteria RNA? You answer, it needs to see a Shine-Dalgarno sequence. You must know this concept. There's pharmacology coming up later. So one more time, a Shine-Dalgarno sequence, shown here by the little dot. It is a sequence of RNA, which encodes from DNA, but it's a sequence of RNA. It's the sequence of RNA to which the ribosome jumps onto the RNA. And so once it jumps onto the RNA, then the ribosome will look for the start codon AUG. That's chapter 4 coming up tomorrow. It's going to read that RNA and making a protein starting with the amino terminus and finishes with a stop with the carboxyl terminus once the stop codon is encountered. Don't worry about start and stop codons. That's chapter 4. But one more time. In order for the ribosome to jump on to the RNA, you answer, it must see a sequence of RNA called the Shine-Dalgarno sequence. What that sequence is, who cares? But it's a sequence of RNA to which the ribosome jumps on, binds, and jumps on, and then it looks for the start codon, making a protein from the amino to the carboxyl terminus. That's gene 1. Other ribosomes jump onto the Shine-Dalgarno sequence of gene 2, starts reading the RNA from the AUG start codon to the stop codon, making another protein independently. Other ribosomes jump onto the Shine-Dalgarno sequence of gene 3, looking for the start codon, and they finish by finding the stop codon. That's next chapter. Another protein is made there. If this were the lac operon, for example, gene 1 might encode the transacetylase, gene 2 might encode the permease, gene 3 might encode the beta-galactosidase, all involved in lactose metabolism. But notice in bacteria, each gene is translated. You must know what that word means, translation, protein synthesis. Each gene is translated independently. All right, is that clear? Okay, again, if you have questions, please use the Q&A pod. Don't worry about repressors. We're not going to talk about the lac operon. Forget about it. Low yield. You're going to be a doctor of eukaryotes, right? And you're going to be a doctor of human beings, not a doctor of E. coli. Who cares about E. coli? Don't worry about gene expression in prokaryotes. It's what's in eukaryotes, which is chapter 5. It's going to be high yield. Don't worry about, I got you, but I'm telling you what you need to know. Here comes something you must know. This is biochemistry integrated with microbiology. Here comes the pharmacology. In pharmacology, what is now if you have the answer in front of you in the book, because you looked at the videos, don't answer. In pharmacology, what is the name of the well-known drug used to treat some microbes, and it targets in biochemistry something we just talked about? What's the name of that drug, well-known drug? Okay, nobody? All right, now, okay, now let me give you a hint here. Now everybody will get it everybody will get it correct. What's the name of the drug when it's administered causes the urine to be red? Everybody will now get it. What's the name of the drug causes the urine to be red? That's right, that is the well-known drug called rifampin. Okay, now what rifampin does is what it targets is transcription, meaning RNA synthesis, and specifically it inhibits the prokaryotic RNA polymerase. So if you were asked, what's the target of rifampin? You say it inhibits the RNA polymerase of microbes. So therefore microbes cannot make messenger RNA, they can't make protein, and they'll be eradicated. Let me ask you this, in pathology, infectious diseases. In pathology, what is the two major infectious diseases in which rifampin is used to treat? What two major diseases? That's right, one of them is tuberculosis. That's right, what's the other one? It could be used for leprosy, it's true, but what's the other one besides mycobacterium? What's the other one? Ah, there we go. That's right. It's used for meningitis. So again, the way they might do, you must know this, high yield. Focus your attention on this, not the lac-operon stuff. Focus your attention on this integration of pharm, path, micro, biochemistry. 4 disciplines here. Because the way they're going to do this on the board exams is, and again, on board exams, they're unlikely to give you a straightforward pathology question. What's the name of this disease? These multi-step questions. What I mean by this is, in the first sentence or two, they might give you all the clinical manifestations that a person has tuberculosis. And they're not asking you, what's the name of the disease? They're not asking you, what's the name of the microbe that causes that disease? They expect you to know, to deduce that that person has tuberculosis. And then they say, then this person was treated with a drug that inhibits transcription. Which of the following drugs was used? You say rifampin. You don't say quinolones, because they inhibit replication. What inhibits transcription is rifampin. Or they might give you the pharmacology, you answer with the biochemistry, or vice versa. Like what I mean by this is, they might say to you, a person has all the symptomologies of meningitis, and then you figure it out, oh, that's meningitis. And then they ask you, this person was then treated with which of the following drugs in pharmacology was used to inhibit the RNA polymerase in Neisseria? You say, well, RNA polymerase that's transcription, therefore it's got to be rifampin, not the quinolones, and not other drugs coming up tomorrow, like the tetracycline, like the erythromycin. They inhibit translation, coming up tomorrow. Here we're talking about transcription. You must get those words straightened out. Replication, transcription, translation. You've seen so far pharmacology that targets replication. Right now you're seeing pharmacology that targets transcription, and tomorrow a whole slew of drugs, antimicrobials, that target translation. You must know what's the target of these drugs. Extremely high yield integration here. All right, now let's finish this off. We're going to talk about now eukaryotic transcription. Now what happens in now before we talk about eukaryotes here, let me say something here. Is you have 46 chromosomal, you have 46 chromosomes. And in each of the 23 chromosomes that you got from your mother and 23 chromosomes that you got from your father, each of the 23 chromosomes, if you added up the total number of nucleotides, it's 3 billion. 3 billion nucleotides in 23 chromosomes total. Now let me ask you this. What percent of your chromosomal DNA in eukaryotes actually encodes for genes? What percent? All of your chromosomal DNA, half of it, or what? What percent of your chromosomal DNA actually encodes for genes? And the answer is very tiny. Only about 1.5%. The best estimate is only 1.5% of your chromosomal DNA actually encodes for genes, which means most of your chromosomal DNA is junk DNA. Now, why is that? Why is that? That's coming up later. There's reasons for this. It's junk DNA, but there's reasons you have so much junk DNA. But only 1.5% of your chromosomal DNA actually encodes for genes. So what that means, therefore, each gene in your chromosomes are very widely spaced. Each gene is very widely spaced. Unlike bacteria, where you have the genes almost side by side or very close proximity to each other, like the lac-operon and so on, in me and you, each gene is very widely spaced. And that means something else. Get ready for a high yield item. That also means each gene in me and you has its own promoter. What does that mean? Those are just words. What does it mean? If you have 20,000 genes, that means you have 20,000 different promoter sequences to which one enzyme, RNA polymerase, has to recognize. RNA polymerase has to recognize what's the promoter for alpha globin, what's the promoter for beta globin, what's the promoter for insulin. It's amazing. One enzyme has to recognize which promoter it is for a particular gene. It's staggering. So what I'm driving at is there's going to be a lot more to the story. That's coming up later on in chapter 5. But the RNA polymerase has to recognize which promoter to bind to, because you have 20,000 different promoter sequences. Now, if you think that's boring, or if you think that's low yield stuff, let me show you or illustrate how the National Board asked this on board exams. According to a student last summer, a student told me on board exams they got a question about osteogenesis imperfecta. He said he knew this patient had osteogenesis imperfecta because the two hallmarks, the fractures that went on and the blue sclera. So he knew it was OI. But they were not asking what's the name of the pathology or what's the name of the protein, collagen, whatever. But what they were asking is, think about this. They were asking, how can the patient end up having osteogenesis imperfecta and there's nothing wrong with the collagen genes? The collagen gene sequences are normal, yet they have OI. That was on the National Board. And the answer was because where the mutation was not in the gene, it was in the promoter for the collagen gene. And so as a result, because there's a mutation in the promoter, RNA polymerase cannot bind to the promoter. It doesn't recognize it as the promoter for collagen. Therefore, RNA polymerase will not bind. You will not make collagen. That was on the National Board. Staggering, right? Yeah, that's what was on you. You have to think about it, and it makes sense. By the way, make sense of it. Oh yeah, that's right. Because RNA polymerase has to bind to the promoter first and then transcribes the gene. And the genes usually have the mutation causing pathology. But what can also cause pathology is the promoter can be defective with the mutation, and RNA polymerase doesn't bind. Amazing. It's not tricky. It's life. It's what happens in real life. This happens in pathology. This is real life situation that goes on. All right, now what's coming up here, just to finish this off, and then we're done for the day, is now there's a couple of terms we need to review, and that's the term called exon and intron. Exon is the expressed part of a gene, and intron is the intervening part of a gene, which means in the RNA being made, not all of it will encode for protein. The information in a gene is fragmented, and the fragments, the exons, have to be spliced together. This is unlike bacteria. Bacteria, they do not have exons and introns, but me and you do. And so these exons, that's the reading frame, have to be spliced together. And because they have to be spliced together, things go wrong, causing pathology. And then we got to ask, well, why do they have exons in the first place? They cause all kind of pathology. The answer is coming up tomorrow. All right, let me finish off this figure, then we're done for the day. Okay, so remember there's 6 items. I'll talk about a few of them right now and the rest of them tomorrow. But the idea is, suppose this represents some gene in the orange and in the brown. So here's exon 1 of a gene, here's intron 1, here's exon 2. So in bacteria, they have operons, but in me and you, there's no such thing as an operon. Why is that? Because of this purple box. Each gene has its own promoter. We do not have operons, or we don't have polycistronic genes, because each gene in me and you has its own promoter. Item number 2, just upstream with those negative numbers is the promoter. And by the way, don't worry about Tata boxes, cat boxes. That's going to be discussed in chapter 5. Right now, we're in the beginning of chapter 3. Don't worry about them now. So RNA polymerase will bind to the promoter, shown in the green. It's going to read the template strand, 3' to 5', making RNA, starting with position 1, position 2, 3. So RNA, remember, you start at the +1 nucleotide, +2, +3, +4, +5. So RNA is being made 5' to 3'. Item number 3, as we'll talk about tomorrow in detail, right at the beginning. There are introns that need to be spliced away that we'll talk about tomorrow. Item number 4, the last item here, is what terminates transcription. Let me ask you this without looking. Don't go looking. What terminates transcription in prokaryotes? If you were asked that question, which terminates transcription in E. coli? That's right. It's the GC stem loop sequence. That's right. But in me and you, some genes have stem loop sequences, many do not. And because of all these exceptions, the National Board has never, ever asked about termination of transcription in eukaryotes. So don't worry about row factors. Don't worry about that low yield item. Don't worry about termination of transcription. So at the end, when RNA polymerase finishes, when it terminates, notice what we got. We got something called pre-messenger RNA. Notice what it has. It has 5' UTR, exon 1, intron, exon 2, and the 3' UTR. Now tomorrow when we come back, we'll talk about how the introns are removed and all these type of things. But one more item, then we're done for the day. One more item. Let me see how you answer this. Suppose integration with pharmacology issues and toxicology and so on. Suppose this weekend, next Sunday, Mother's Day. Suppose next Mother's Day we don't have any lectures. And so you want to think about all this biochemistry that we talked about in the previous week, next Sunday. So you want to go, you get in your car and you want to be alone. You don't want to be dealing, you want to do no hassle, you want to be alone. So you get in your car and you drive to the nearest park and you want to walk around by yourself and think about all this biochemistry that's finally making sense. And because you're in such a hurry to go into the woods and walk around, you don't eat anything. You don't stop at McDonald's to eat. So as you start to walk around, you're starting to get very, very hungry. Oh no, I should have stopped at McDonald's because I'm so hungry. And then you look, and in the distance, there's mushrooms. So you go over there, you pick those mushrooms, you eat those mushrooms, and you kill over and die. Here it comes in toxicology. What killed you? Why is it dangerous to eat certain poisonous mushrooms? What's in the mushrooms that can kill me and you? That's right. That's right. It is alpha-amanitin. Alpha-amanitin. So here comes the question. What does alpha-amanitin do to kill? In biochemistry, what is alpha-amanitin targeting that can kill me and you? That's right. What it does, it inhibits the RNA polymerase in eukaryotes. Therefore, it cannot make proteins, and therefore you can die under those conditions. By the way, in my opinion, don't worry about the various RNA polymerases, RNA polymerase 1, 2, and 3, which one makes messenger RNAs or tRNAs. Low yield, it's in your book and all that. Don't worry about that. Focus your attention on things like alpha-amanitin, this issue of OI, each gene has its own promoter. Focus your attention on what you need to know. Alpha-amanitin is an inhibitor of RNA polymerase in eukaryotes. In other words, alpha-amanitin is a lot like rifampin in prokaryotes. Rifampin, alpha-amanitin, one's prokaryote inhibitor of RNA polymerase, the other is eukaryotic RNA polymerase inhibitor. So again, you must know that issue here. All right, so we're done for the day. Now let me say, so tomorrow we'll come in and talk about what happens to the pre-messenger RNA. You don't call it messenger RNA yet. You call it a primary transcript, also called pre-messenger RNA. Now, we're done. Now, what we're going to do is on the Facebook, now those of you, again, hopefully you signed up for Facebook. You should have. I'm going to post, tonight, I'm going to post 3 questions. I usually give 3 questions after every session. and I post them, and you can always message me there with the email there on the Facebook. But anyway, these questions that I post, don't just put option E or option A. Don't do that. I want you to explain your answer, defend your answer. That's how you learn. If you put option E, that does nothing. It does nothing. I want you to explain. I noticed when I was watching some of the other people, other instructors, a lot of you were just putting option E, option A. It does nothing. Explain your answer, and then after you type in your answer, look at other people's answers, and then if you need some help or whatever, explain, interact with each other. That's part of the learning process. If you have to explain, you remember it a lot better, because when you make a mistake, you say, oh yeah, that's right, I should have known that. Explain the answer. Don't worry if you give the wrong answer. You're among friends, so you give the wrong answer. I gave many, many wrong answers in my lifetime. If you give the wrong answer, who cares? But this is part of the learning process, which I'm trying to get you to do. So don't type in option A, explain things. It's for your benefit. It's for your benefit to go through these questions. So I do 3 questions on average after every session. And then by tomorrow, sometime tomorrow, I hope to get you I'll explain the answers or point to somebody and say, look at so-and-so's, they explain it better than me, something along that line. But please explain the answer as part of the learning process. Okay, everybody, we're done for the day. I'm looking forward to seeing you. So tomorrow we're going to finish off chapter 3 in transcription, and then we'll get into chapter 4 of translation, which takes a long time to translate. Be ready for a lot of pathology and pharmacology. And then after that, we may get into chapter 5 of regulation of gene expression. Okay, everybody, you take care. Have a great evening and great day tomorrow, and I look forward to being back to you. Hopefully it's been beneficial. Hopefully you're learning and not memorizing. You're understanding and not going through mnemonics. Okay, everybody, have a great evening. And again, my email is on the Facebook site. You can see the Kaplan, Dr. Turco. You'll see it there on the messages. You'll see the Kaplan site there. Okay, everybody, so you take care, and I'll see you tomorrow. Take care.